Hi, everyone. Welcome back. I hope you all got a chance to check out some of the poster presentations during lunch break. For our second faculty speaker of the day, I would like to introduce Dr. Taji Harris. Dr. Harris is an associate professor in the neuroscience department at the University of Virginia School of Medicine. She is also a member of the Center for Brain Immunology and Glia at the Carter Immunology Center. She received her PhD from the University of Wisconsin, where she began her scientific career studying parasitic pathogens that infect the central nervous system. Dr. Harris and her laboratory currently research the intracellular protozoan parasite Toxoplasma gondii. The Harris Laboratory aims to identify factors within immune-privileged tissues that influence local resistance to Toxoplasma gondii. Today, we have the pleasure of listening to her talk titled Alarmin, the Immune System in the Brain, T. gondii Parasitic Model. Please welcome Dr. Taji Harris.
Thanks so much for that introduction, Lauren. It's just a pleasure to be here virtually. So my background is the lawn at the University of Virginia. But I do have very fond memories of Philadelphia. I did my postdoc at Penn. So that was a great introduction and overview of my research and where our perspectives sort of coming from. So it's really a pleasure to give talks to infection and immunity groups. I am in a neuroscience department, so I usually speak to really diverse audiences, but it's really great to get to the heart of immunity in the brain. And so that's what I'm going to focus on today. A lot of what my lab's really trying to understand is how do we build an immune response in the brain? And this is just different than any other tissue. And so I'll get into a few of those details, tell you a little bit more about toxo, and then get to a story about the alarmin IL-33 and how we think it's working in brain to promote immunity to this pathogen. Okay, here we go. So building an immune response in the brain is different from other tissues. And in the textbooks for the longest time, we said, the brain is immune privileged, so really immune reactions are not happening. And now I think we'd revise that for immune privileged tissues to say that immune reactions occur, they just occur differently. And so we're still really behind in trying to understand all of the features of these tissues that help to promote or limit immune reactions. And so there's a lot of research to be done. And there are several factors that make immune responses in the brain different. And one thing I would just characterize is that they're generally slower. So you can transfer cells that are specific for brain antigens, and it takes them many days to arrive in the brain. And this is something we just really don't understand. One of the hurdles is that any immune cell that wants to enter the brain to combat a pathogen or enter for any reason has to cross the blood-brain barrier. And essentially, we probably all have some concept of what the blood-brain barrier does, and it's to keep things out. And one of the ways that it does that is that there's two layers of basement membranes in this diagram. The endothelial cells here will put down a layer of basement membrane, but also these astrocytes, these sort of structural cells of the brain, also put down matrix. And so these cells have an extra feat. Not only do they have to be activated enough to cross over the blood-brain over through the endothelium, they then have to be able to chew through two layers of basement membrane before they even reach the deep brain tissue. So we're already asking a lot of the immune cells, but there could be great advantages to limiting immune reactions in the brain. And therefore, the blood-brain barrier could be very protective of the brain. So limiting immune reactions in the brain is probably a good thing on most levels. There are a couple other features of the brain that make immune reactions different and slow, and that's there are no classic lymphatic vessels in the deep brain tissue. So recently, the lymphatic vessels were described that run in the meninges and the outer layers of the brain. They run right down the center of the brain along the major dural sinuses. And so basically, if any antigens are going to be able to leave the brain and then activate the immune system, they've got quite a journey in order to do so, and that might be what leads to the immune reactions being somewhat slow. There's also very low MHC expression in the brain at baseline. So if you do a classic stain for class 1 or class 2, the levels are very, very low. But with inflammation, they can be upregulated. But at baseline, the brain is a very quiet place. And these could be all the reasons why immune reactions are pretty slow to develop in the brain and quite tricky to develop too. So what my lab does is uses an infection to try to understand how immune reactions happen in the brain. in a condition where it's very important for the immune cells to respond. And for the most part, the immune system responds without doing much damage to the brain, which is a positive. And so to study this, we're studying Toxoplasma gondii. As Lauren mentioned, this is an intracellular protozoan parasite. So it has to live with inside cells. It's a eukaryote, so it's more like us than bacteria are. And it's sort of a fascinating disease with a lot of prevalence. So up to a third of the world is infected with this parasite, but we don't hear much about it. And the reason we don't hear much about it is that our immune system does a really fantastic job of controlling this bug. And so what people develop is a long-term chronic infection with this parasite. So there's really no evidence that people ever clear this pathogen completely. And so you need constant immunity to keep some pressure on the pathogen so that it doesn't cause problems. So there are susceptible patient populations. Those include people with AIDS or transplant recipients and some people undergoing chemo. If the immune system is quite suppressed, then Toxoplasma comes out of its latent state and causes a lot of issues. And so it's for the reason that most people don't develop symptoms that we're quite interested in Toxoplasma. This means that the immune system is doing a wonderful job of keeping this pathogen at bay, a pathogen that ends up in the brain. And so we can learn how an immune response functions well and effectively in the brain by studying this pathogen, which is a natural pathogen of mice as well as humans. Okay. So I love this image for illustrating the basically the dissemination of Toxoplasma post-infection of a mouse. And so what our colleagues at Stanford did was generate a bioluminescent parasite. So Toxoplasma is quite genetically tractable. And so they used an IVUS imaging system to really just watch where the parasite disseminates over time post-infection. And that's what the number corresponds to in this upper right corner. This is just the day post-infection. And so what you can appreciate is that the Toxoplasma spreads throughout the body. And so during this first week of infection, and they here injected the parasite's IP, as I will do as well in these upcoming experiments. But essentially the parasite spreads throughout the body. And then you can notice between day 10 and 12 here, this is when you get a big change. This is when the adaptive immune response kicks in. And so the B and T cell response here clears the majority of the parasites from the body. But as you go further and further out to this chronic phase, which really is from about 3 weeks on post-infection, the parasite's largely just within the brain at that point. We do know that it's in other immune privileged tissues. We can find it in the retina as well. But essentially, the parasite's largely found within the brain during this chronic phase of infection. Okay, and so these images really nicely demonstrate how potent the adaptive immune response is at clearing toxo. And so many studies have been done. And so we really benefit from a wealth of knowledge that's been gained by studying the immune response to toxo during these first 2 weeks or so. So we know a ton about this. And so essentially what we know is that dendritic cells are key sources of they're both antigen presenting cells, but they also make a lot of IL-12 that helps to prime this polarized Th1 response that ultimately leads to the production of interferon gamma. So interferon gamma is the key cytokine that mediates immunity to toxo, and it does so via multiple mechanisms, but essentially cells that respond to interferon gamma can mount several antiparasitic immune responses that are all induced by interferon gamma. So not every cell type responds in the same way, but basically interferon gamma will stimulate one of many mechanisms that end up killing intracellular toxo. So this is what we know about the acute phase of infection, these early time points, but we're more interested in what's happening to control toxo once the parasite's in the brain. And so through multiple studies, we know that T cells are still very important. Interferon gamma is incredibly important. But we're trying to understand then basically how you mediate immunity to the brain when you have the parasite present. And so more of these questions will become evident as we move forward. So a lot of our questions really came from looking at images. And so my lab uses a lot of imaging techniques to try to understand where the parasites are located, how the immune cells enter the brain, how they move through the brain, and then how they end up finding the parasite. And this becomes relevant for a few reasons. But here, this is just a really simple confocal image. So this mouse has been infected for 4 weeks, and we've sectioned the brain and stained for toxoplasma. And so this structure up here in the upper right, this is a parasite cyst. This is the dormant phase of the parasite. So what happened here is that a single parasite entered a cell, created a parastaphus vacuole, multiplied a bunch of times, and then created a cyst wall. This entire structure is still intracellular, which is really quite amazing. So we will see cysts that have diameters of 30 to 50 microns. And so these are giant structures for cells to accommodate. And so one thing we notice is that we definitely have cysts in the chronic phase of infection throughout the brain, but then we also notice these regions, and these are less common than cysts, but not uncommon at all. And in these regions, what we have are little individual parasites. And so what's happened here is that these cyst forms have reactivated and these individual parasites have come out. And so all of these parasites are capable of going on to infect and invade new host cells. But what is readily apparent to us from this image and other types of images that we've taken is that there's a really profound cellular immune response to the parasite here. We're just seeing this by appreciating this by just the DAPI staining. But this is almost granuloma-like. It's not a classical granuloma in any way, but we've got a huge infiltration of immune cells specifically to this location where the parasite has reactivated. So this has raised a lot of questions as to what's going on in these specific locations in the brain. While we don't understand this completely, a lot of our imaging suggests that we don't get many immune cells recruited to areas where we have parasite cysts. So we think that these cysts might be somewhat quiet or able to hide from the immune system because they are still intracellular and largely within neurons. So this raises really interesting questions about how the cyst form of the parasite sort of stays under the radar, which is in vast contrast or stark contrast to when the parasites have reactivated and are actively able to replicate it in the brain, we get a really strong immune reaction. So we're trying to understand what the difference is, and then ultimately how important it is for the immune system to recognize this reactivated parasite region. Okay. So what we wanted to do is just zoom in on these areas. And so we call these various things like inflammatory foci or regions of reactivation. But what they all have in common is that the parasite is basically able to invade new cells. What we see in these regions are huge accumulations of immune cells. So here we've stained for what are largely myeloid cells, or sort of the macrophage lineage. And so neuroimmunologists use this marker IBA1. It's a surface marker of basically myeloid cells. So it'll pick up microglia and blood-derived macrophages. And there's a lot of class 2 expression. So this is just showing that we're getting this is just a massive accumulation of cells surrounding the area where the parasites have sort of woken up. We also get T cells coming into this region. It's not just the myeloid cells. And so we're like, this is fascinating. So they seem to congregate in a very specific region within the brain. And so we just really want to know more about that. And so one of the questions we have is like, so which cells are congregating? This IBA1 marker cannot delineate between our sort of tissue resident microglia and the monocyte-derived macrophages that would come in from the blood. And so why is this an interesting question? Well, so basically we're asking, is it the microglia or the macrophages? And so microglia are interesting cells and have absolutely basically captured the imagination of most neuroimmunologists. So we want to know far more about them for the very reason that they are the sole immune cell that's in the deep tissue parenchyma of the brain, right? So we have this blood-brain barrier. The immune system doesn't just circulate through the brain to detect problems or pathogens, invaders. We really are reliant on microglia, the cells that are in the deeper tissue. Microglia are interesting because they have really early origins during development. So they develop from the yolk sac and then traffic to the brain, and then this blood-brain barrier sort of seals them in the brain, and then they self-renew. So most of the other tissue resident macrophages throughout the rest of the body over time do get replaced by cells from the bone marrow. That is not true with microglia. So they're kind of a unique tissue resident cell with unique properties. That's in contrast to the other population of myeloid cells that we have in the brain, which are derived from monocytes. So basically, these are the cells that are coming out of the bone marrow, traffic through the blood, and then come into the brain. And so using RNA-seq, we have found that these two populations, the resident microglia and the blood-derived cells, have pretty distinct effector capacity. So we really wanted to know which cells were present in these clusters and surrounding parasites. And so one of the tricks we used was just to create a microglia reporter system. One of the issues with trying to study microglia is that they share almost all common surface markers with the blood-derived cells. So you got to be creative in trying to label the tissue resident macrophages. And so the system that we've used, which works quite well, is an inducible Cree system under the CX3-CR1 promoter. So that hits most tissue resident macrophages throughout the body, including all of the microglia. And so we've got it on a AI6 Cree reporter mouse. So when there's Cree activity, the cells become green. And so what we do is we treat with tamoxifen when the mice are fairly young. We label all of the macrophages throughout the body, including the microglia, but all of those peripheral populations, so all the populations down in the brain, do get replaced by cells that are in the blood. So essentially, after we wait for 4 weeks, all of our microglia stay labeled, whereas the cells in the peripheral tissue have turned over and they're no longer green. So basically, we give a dose of tamoxifen, label all the cells. There's a lot of turnover in the periphery. And then 4 weeks later, we have basically the microglia, the only remaining green cells in the body. So using this system, we're able to even do 2-photon microscopy. So these are live animals where we've been able to thin down the skull and peer through the skull and see how these cells are behaving. So microglia are really fascinating. They have these long processes that they constantly wiggle through the tissue. This is what they look like in a healthy animal. Post-infection, they take on a very different shape, and maybe you guys can appreciate that here. So instead of having the long spindly processes, they almost start to look like crustaceans. Their processes become much thicker and they stop moving as much. So this is just a very, so we're only able to look into the brain a couple hundred microns. And so what we're able to see, though, is that the microglia in the brains of these infected animals are definitely activated. They're responding to something, but we're quite a distance away from where we would have cysts form or have cysts reactivate. And so we wanted to use a different type of microscopy to understand if the microglia were actually going to mobilize towards regions of parasite replication. And this is what we found. These images are quite striking. They're from my student, Maureen. And so here she's been able to stain for the parasite. In red, ME49 is just the strain of parasite. And what we find is that these microglia here that are green, so they're looking quite unhealthy, the microglia from infected mice, and especially in this region. They're very puffy. They're very vacuolated. They look pretty shredded, to be honest. So the microglia up here in this corner is a little bit more like a normal microglia. It's even looking a little bit stressed out, but these microglia are definitely moving, we think, towards these regions where there's parasite replication, because there's all of this dark space surrounding the region where the parasites have reactivated. Microglia do a really fascinating thing where they normally tile throughout the tissue and have their own territory, so they don't overlap with their neighboring cell. But here we're seeing when they do respond to this insult in the brain, they are congregating. And one of the other cool things about using this tool is that when we use the IBA1 antibody, we can then, that still stains the microglia, but it also stains all of these cells that have come in from the blood. So all of the cells here that are just white and not green, those are the ones that have come in through the blood. And so we can see that we've got both cells that originate from the brain, the microglia, and the macrophages coming in to surround these regions where we've got parasite that needs to be contained. Okay. So then our next question really became, well, what's going on in these regions? So we know we can recruit in T cells and myeloid cells. What else is happening in these regions? Why are the cells congregating specifically, right? So we can say, yeah, there's parasite there. They're probably responding to the parasite, and that could be. But we wanted to dig a little deeper and just say, okay, what's happening in these regions? And so in all of these images, we're basically looking at the inflammatory cells or all of the densely packed nuclei in the center. And so that's just DAPI stained. And so these are different regions in different animals, but essentially they're all examples of these inflammatory regions where toxo is reactivated. And so when we start to stain for brain resident cells, so we're staining here for oligodendrocytes using the marker OLIG2. GFIP is staining the astrocytes, the sort of structural cells of the brain. And even when we stain for neurons, in these regions where we've got a lot of inflammation, these brain resident cells are missing. And so we're really wondering what's happening here. Sometimes what we think is that the inflammation might push these structural cells of the brain out of the way. That's a possibility, but we're not really seeing an accumulation of these cells on the borders. We're actually seeing that these cells look very sick. And so we're wondering if they're actually dying. And so that's what we do hypothesize is happening, is that in these regions of inflammation, the brain resident cells appear to be very sick or ill or dying. And so studying cell death in a tissue is really, really difficult because we think that cells, once they start to get sick, are cleared very quickly. So it's sometimes difficult to catch cell death in a tissue. But one of the ways, and there are several ways, is you can look for canonical markers of cell death. So by tunnel staining, you can pick up basically fragmented DNA. And so we do see tunnel staining in these regions where we've got the loss of astrocytes. So the astrocytes are staying red here, but they're absolutely missing in the middle of these lesions, and we've got some evidence of cell death. We also see cells that take up propydium iodide, which basically means they've lost membrane integrity. We also see some evidence of ASC spec formation, so that may be activation of the inflammasome, and also some form of pyroptosis going on. So we've got some evidence that there's some cell death going on in the brain. And so we started to think about this. We're like, okay, so we really want to know how the immune system's being called into this specific area of the brain. We seem to have some cell death and loss of our brain resident cells. Could that be the trigger of what's bringing immunity to these locations, or the immune system to these locations? And so we started thinking a lot about alarmins. Alarmins are super cool molecules. They've got a really appropriate name in that they tend to send out the signal that there's been damage to the tissue. And so alarmins are molecules that are all preformed in cells. And so a lot of times when we think about an immune response, we're thinking about response to something and then the transcription and translation of new cytokine that then gets secreted and starts to alert other cells. The alarmins are going to be a bit faster because they're already preformed. So healthy tissues have high level of alarmin expression. And the reason that they don't normally activate the immune system is that they're largely kept within the cell. They're not typically secreted. And so a couple of examples of these molecules include IL-33, what I'm going to tell you about today. Also IL-1 alpha. My lab has a story about IL-1 alpha that we published this summer, which is somewhat parallel to the story I'm going to tell you. But other molecules include ATP and HMGB1, among others. And so essentially, when cells undergo some sort of aberrant form of cell death or there's damage, these molecules can be liberated from the cells and then detected, and then you can build an immune response. So this is what we were wondering. Is this happening in the brain? Is this part of what the destruction that's mediated by the parasite, is this how it's sensed? And this is how the immune system knows where to go within the brain. Okay, so I'm going to tell you a little bit more about IL-33. It's a really cool molecule. I know several people in Philadelphia work on IL-33, and a lot of work has been done on it. A little less work has been done on the brain, but there's a ton of IL-33 in the brain. IL-33 lives in the nucleus of cells where it's quite unclear whether it has a function in the nucleus or if that's just where it's localized. But once IL-33 is liberated, it can bind to its receptor that is composed of ST2, and the co-receptor is IL-1 receptor accessory protein. So the unique receptor for IL-33 sensing is ST2. So in a lot of the studies I'm going to tell you about in upcoming slides, I'm going to use ST2. But essentially, the recognition of IL-33 looks, with the downstream signaling, it looks a lot like toll-like receptor signaling, or basically activation of an innate immune system, innate immune response. So IL-33 is going to signal through MyD88 and essentially activate a bunch of production of a bunch of molecules that can help facilitate an immune response. And so a lot of what we know about IL-33 is in the context of type 2 immune responses against sort of helminths in the guts or lung or allergic responses. And so we're talking here today about toxoplasma, which is really a canonical Th1 inducing pathogen, and the brain. So it's sort of a different context of how IL-33 is important in another tissue. Okay, so like I said just before, their brain is full of IL-33, and this is really quite interesting, like big questions as to why. So here we've stained for IL-33 in red, and you can appreciate the nuclear localization of the IL-33. And we find that the IL-33 is expressed largely by two cell types, and this is prior to infection and during infection. This really doesn't change much with the presence of inflammation. But the astrocytes here, so astrocytes are support cells. They give structural support. They also help to keep neurons healthy. They're here in white. They do express IL-33 largely in gray matter. And then in the white matter, it's a lot of these myelin wrapping oligodendrocytes, those that sort of insulate the nerves, the neurons. They're expressing a lot of the IL-33. So there's some regional difference, but basically there's many cell types within the brain that express the IL-33. So when we looked during infection, we found this really cool pattern that you can now appreciate. So with IL-33 staining, how much of the tissue has IL-33, how many cells are expressing it. But in the center here, this is a region where the toxoplasma has woken up and is causing some problems. We get a couple of remaining parasites here. We can see that we don't have IL-33 reactivity through the center of this area. This could suggest that some of the IL-33 expressing cells are dead and could have liberated their IL-33. But it does raise the question of whether or not sort of the absence of IL-33 suggests that it could be signaling. And so this is really quite suggestive, and it's very difficult to measure the local release of cytokine. As a lot of you probably do appreciate that we can easily measure cytokine in supernatants once we get a huge concentration, but to try to measure local concentrations of cytokine within a tissue is really quite difficult, especially if you try to homogenize a tissue like brain. You're going to end up releasing the endogenous IL-33, which is really problematic. And so in order to really know if we're getting release of IL-33 in response to infection, we decided to measure IL-33 in the cerebrospinal fluid. And so essentially, this is the fluid that bathes the brain. And so if there's going to be the release of IL-33, we thought we could detect it here, and amazingly we could. So these are really kind of heroic experiments undertaken by Igor Smirnov and my student Kate Still to pool CSF from many animals to be able to have enough to run an ELISA. And so we could detect that IL-33 is indeed liberated during the infection. Okay. We had some other questions too about whether or not this would be relevant to human disease. And so we wanted to stain some healthy human tissue just to see if IL-33 is expressed similarly in the human brain. And it is expressed by astrocytes. So these are astrocytes here, also marked by GFAP. They're just much larger in humans, and they are expressing IL-33 like in the mice. We were able to get a hold of a few human samples, and what we saw from people with toxoplasmic encephalitis And what we saw in healthier regions of that tissue, we could see nice IL-33 signal from cells, but when we have presence of a lot of parasite and a lot of destruction in these areas, we're no longer seeing the presence of IL-33, which suggests that also in humans, there could be the liberation of this cytokine, which would make it relevant for inducing local immunity. Okay. So we have the cytokine present. Is it important? So the way we wanted to first ask these questions is just by looking in the ST2 knockout mice. And these are basically lacking the specific receptor for IL-33. And what we saw, so we infected mice for 6 weeks. And what we saw is that in mice that lack ST2, we have far more cysts in a region. And sort of typically what we see in wild type mice is that we'll see the formation of these toxoplasma cysts. They're kind of alone, right? So we'll see one in a field of view. And often when we see clustering of multiple cysts, this is indicating that we're getting some sort of poor immune response that's allowing multiple cells within one area to get infected. And so when we quantify the number of parasites we have like about a threefold increase in parasites. And so this deficiency in IL-33 sensing is not enough to make the animal succumb to infection, but they just have a difficulty handling the parasite within the brain. So they're doing worse, but it's not fatal. Okay. So we wanted to dig into what's happening. So we've got the suggestion that we've got impaired immunity when the mice can't sense IL-33. And so the way our lab just started to address this question is to do some really, really basic flow cytometry. And so we've done more sophisticated gating, but this sort of encompasses everything that we were looking at. So if we take cells that we purified from the brain and stain them for CD11b or CD45, the microglia sort of conveniently fall into the CD45 intermediate gate, whereas the cells that are coming in from the blood are in this CD45 higher gate and both stained positive for CD11b. And then most of our cells that are 45 positive but 11b negative are largely T cells. And so when we just look at this very generic overview of the immune system, what we really saw is that we were lacking these CD45 high, CD11b positive cells. And so these would correspond to cells that were monocytes in the blood, came into the brain and became something else. And so we wanted to dig into this further. And so one of the things my student Kate did is generate these really striking images. So what she's done here is taken a CCR2 RFP reporter mouse and just false colored them green. That's why they're not red. But and so what these mice do normally is report the entrance of basically monocytes, so a CCR2 expressing cell. And so if we were to look at a naive animal that is a CCR2 reporter, we might see 3 or 4 dots. But this infection with toxoplasma leads to a really, really robust immune response, and you can appreciate that here. We've got lots of infiltration into the choroid plexus, the meninges, and into the deeper tissue parenchyma. But when we crossed these reporters onto the ST2 knockout background, this just got quieter. So this was very much in line with our flow cytometry data that the absence of IL-33, we're signaling we're basically seeing a reduction in the recruitment of monocytes into the brain. So we started to think about, well, what would cause something like this? So we just went back to our sort of view of how we actually try to get immune cells into the brain. And there are a few requirements. So cells have to be the immune cells have to be quite activated and expressing integrin receptors, so they can interact with an activated endothelial layer. And so we know that VCAM and ICAM are really important adhesion molecules on the blood vasculature that needs to be upregulated in order to get these cells even to stick. And then once the cells sort of traverse the endothelial layer, we believe there's a role for chemokine, and there's some good evidence that there's a role for chemokine in bringing these cells deeper into the tissue. And so what we did is just a really generic quick screen using sort of whole brain RNA and then doing real-time PCR just to see whether or not all of these factors that we know are involved in bringing immune cells into the brain, whether any were like sort of expressed at lower levels in the absence of IL-33 signaling. And what we saw is that a few molecules were down, including CCL2. And this is the chemokine we heard a little bit about this morning that's really important for bringing monocytes into tissues. So this fits really well. We've got a defect in bringing these cells into the brain, and we've also got a defect in the ability to make the chemoattractant that brings the cells into the brain. We're also missing a few other, or had reduced expression of CXCL10 and CXCL1. And one thing that we know from the literature and a bunch of other studies is that all three of these chemokines are made by astrocytes. So this was suggesting to us that perhaps our astrocytes within the brain are making less chemokine and are likely less activated. So when we did a similar approach to look at CCL2 in the brain, and so again, these are sagittal sections of brains of infected mice that were then stitched together. And the CCL2 reporters are quite stunning in that we can now see the very focal patches of chemoattractant production that would help to bring monocytes into the brain. And so just like our real-time PCR data was telling us, there's a reduction in the amount of CCL2 when the mice lack S22. And so a few things caught our eye. When we look at the brains of toxo-infected animals, we see the activation of cells throughout the brain. But the CCL2 reporters were showing us some things unique that we really hadn't observed very well before, is that there are specific pockets and regions in the brain where CCL2 is being produced. And so this might dictate some of the regional accumulation of immune cells within the brain. And again, the production of CCL2 is not completely gone in the absence of IL-33 signaling. There's just far less of it. So there's clearly other factors that are driving CCL2 production, but IL-33 is one of them. And when we look for which cell types are making the CCL2, it's 80% of it is coming from the astrocytes. And so this is a local structural cell within the brain. This is not typically what we would consider an immune cell, but these astrocytes are very prone to reacting to inflammation. And so they seem to be a critical source of CCL2, which I'll come back to later. Okay, so we have reduced chemokine levels. We also have reduced functionality of the cells that do come into the brain. So one of the things we know is that the monocytes that come into the brain from the blood will upregulate the ability to make nitric oxide. And this is a potent antimicrobial molecule. So basically, iNOS is the enzyme that ends up making nitric oxide. And when we look at our cells in the IL-33 receptor knockouts, they just make less nitric oxide. So the cells that are coming in aren't getting fully activated and wouldn't be as potent to end up killing toxoplasma. And that could explain why we have a higher parasite burden in the ST2 knockouts. Okay. And then this is sort of really dramatic when we look at it by confocal imaging. We can stain for the iNOS enzyme. And so what we usually see are these very clear and distinct patches of iNOS production within the brain, exactly where the parasite is replicating. So there's quite an ordered process to get these cells to a specific location, activate them, and then they're making the key factors that can control toxo. But when the mice lack IL-33 signaling in these ST2 knockouts, we still saw some iNOS production. It was just these regions were much smaller. So that's suggesting that some other factor is kicking off these inflammatory lesions, but IL-33 is helping to make them larger and more potent. And so there's a lot more work to be done to knowing all the factors that initiate focal immunity in the brain, but IL-33 is definitely a potentiator of that inflammation. Okay, so just to summarize what I've shown you thus far, we've got some evidence that in regions where toxo is reactivated and actually, invading cells and not in its quiescent form, we're seeing some degree of tissue damage or death that we don't fully understand. But we believe that this is what's releasing the IL-33. And then what we're finding is that IL-33 is potentiating CCL2 production, which we think is largely from astrocytes, which would help to bring cells into the brain, mature them, and make them actually able to kill toxoplasma. But we had a few questions in here questions that have sort of eluded um, the neuroinflammation crowd, and trying to understand which cells in the brain are going to really sense the IL-33. So we know that astrocytes are making the CCL2. Is this direct? Is this a direct action of IL-33 on astrocytes or not? And then really also, how important would CCL2 from just the astrocytes be? So those are the two questions that I'm going to address in the upcoming slides. So one of the cool things about studying IL-33 in the brain is that it's definitely functioning quite differently than how we understand IL-33 functions in the rest of the body. So a lot of what we've known from sort of the type 2 immunity field is that IL-33 is expressed in the epithelium of lung and gut, and if it's damaged through various mechanisms, that can lead to the release of IL-33 and then the initiation of type 2 immunity. And a lot of the sensing of the IL-33 is done directly by immune cells. But when we're thinking about what's happening in the brain, just because of the presence of the blood-brain barrier, if we were going to release IL-33 from the astrocytes or the oligodendrocytes, in a normal situation, the immune cells are very far away. We're not sure that they could sense it. And so we were really wondering, what's a key cell type that must react to IL-33 in the brain to potentiate immune responses? And so basically, we were thinking there could be a couple candidate cell types within the brain, or it could be immune cells, because we do have the presence of mast cells and some and Tregs and some ILC2s in the brain. They're just sitting up in the meninges, very far away from the deep brain tissue. So we started by doing a really sort of brute force approach of trying to understand if the immune system was important at all by doing a bone marrow chimera. And so this is a really detailed slide, but essentially what we wanted to do was generate mice where the immune system was either wild type or ST2 deficient, and try to ask, is that important or not? Or is it more important that the non-immune cells are ST2 sufficient or not? And so we irradiated mice, did bone marrow transfers, and the way to read these graphs is that the donor is listed first. So the immune component is first and the recipient is second. And so once we infected these bone marrow chimeras, what we found is that we had defects in the ability to recruit these INOS expressing monocyte-derived cells in mice where their recipient was ST2 deficient. So it didn't matter what the immune response was or what the immune system was capable of. It was whether or not the radio-resistant cells or the sort of tissue-resident cells expressed ST2. So essentially, the data we have when we look at multiple cell types and parasite burden is that this doesn't implicate the immune system. It suggests that a brain-resident cell is responding to the IL-33. Okay. And so we started to go down the road of trying to figure out which cell type is most important. And one of the fascinating things is, based on the literature and also what we found, is when we look in naive animals, the majority of the ST2 expression or the IL-33 receptor expression is on microglia, which maybe makes sense, they're the immune cell of the brain. But once we infect the animals, the cells that express 11B stop expressing ST2. And we don't know what causes this change, but a similar change is also seen in the lung. And what we see happens is that now we've got astrocyte, these ASCA2 positive cells start to express ST2. So they express low levels prior to infection or inflammation, and then they start to express higher levels. So this basically led us to say, well, it could be microglia that respond early, but perhaps there's also a role for astrocytes in sensing IL-33. So my student Kate started to make all of the Cre-Lox mice to try to address this question. And so we were able to delete ST2 from specific cell types using Cre. And so basically, the first thing we did was use a CX3CR1 Cre. This is a constitutive Cre. So it's going to hit basically tissue-resident macrophages throughout the body and a few circulating monocyte populations. But essentially, when we examined these mice, they had mild defects, but for the most part were really okay, and there was really no change in parasite burden. So this was saying, okay, it's probably not the microglia that respond to IL-33 in the brain. So what about astrocytes? And so what we found in these experiments is that when we used a GFAP cree, and this is going to hit the majority of astrocytes, but we also compared them to the full body ST2 knockout. We had all of the same defects in cell recruitment as the whole body knockouts and an increase in parasite burden. So this is suggesting that astrocytes are indeed a responder to the IL-33 in the brain, which was great to show because we didn't know that before. But the other question we had was, okay, so the astrocytes seem to be making CCL2 in response to the infection. So how important would it be that one cell type makes one chemoattractant? And so the CCL2 reporter mice that we had were also floxed. And so we just took the opportunity to get rid of CCL2 production from astrocytes specifically. And our results were somewhat surprising to us that we didn't expect such a strong phenotype. But what we found is that the astrocytic production of CCL2 accounted for a lot of the recruitment of monocytes, and we had dramatic increase in parasite burden in these mice, which is showing that astrocytes are key responders to local damage signals and then key producers of chemoattractants that help to bring the immune system to specific locations in the brain. Okay, so that's essentially what I just said, but I want to summarize it again here, that we've got some open questions as well, but we know that when the parasite's replicating within the brain, there's some sort of damage being caused, and we don't really know what's releasing the IL-33 in this system. We know that the IL-33 is really important for acting on astrocytes for them to make CCL2. And then that CCL2 is quite important for drawing these cells into the tissue parenchyma, helping to fully activate them, which we think then is leading to antiparasitic effector capacity that limits parasite replication within the brain. Okay, so I just want to wrap up with the acknowledgements. So this work was done by my student, Kate Still. She defended her PhD 2 days ago. So she's a newly minted doctor. And so the majority of the work was done by her. Maureen Cohen also did the microglia work. She was also assisted by past members of the lab, Sam Batista and Carly O'Brien. I also want to acknowledge Aaliyah Tate-Voino for the use of IL-33 knockout mice. And I also want to give a shout out to our Center for Brain Immunology and Glia at UVA. So this is a really unique grouping of scientists. We're all really interested in neuroimmunology, really fun place to work. There's lots of job openings if anybody's on the career hunt, and I'd love to take any questions.
Okay, so our first question comes from Dr. Vincent Tam. Said, beautiful talk. What is the mechanism for the necrotic cell death? Is it pyroptosis or necroptosis? Do the different programmed cell death pathways release IL-33 differently?
Great question. So we've dug into this. I'll tell you what we know. It's complicated. So I think one of the biggest hurdles to addressing this question is that when we're working in a tissue, it's hard to detect the release of cytokine. We might have to measure it in CSF to really definitively show that one particular type of cell death leads to the release of an alarmin. So what we do know is that RIPK3 deficient animals have a very mild phenotype, if anything. So we don't think there's a huge role for necroptosis in this system. We do have some evidence for inflammasome activation. Basically, ASC specs form specifically in microglia in the brain. Microglia don't express IL-33, but they do express IL-1 alpha. And so we've found in another study that pyroptosis, or at least Gas-Dermin-dependent pore formation, leads to the release of a different alarmin. So we really don't know how IL-33 is being liberated at this point. we're going through different death cascades and different types of initiators of cell deaths to really try to understand that. We haven't ruled out that the parasite itself is triggering cell death in these astrocytes. That's been really hard to pin down. We might have to visualize that using two-photon microscopy, which has its own challenges, but we really would like to observe the sort of timing of cell death as these cells are coming in closer contact to the parasite to see if the parasite's mediating cell death. But that's a great question.
Our next question is from Cassandra Spector. She said, thanks for a great talk. I'm wondering about the effect of IL-33 release on astrocyte foot process integrity near the blood-brain barrier that might affect monocyte infiltration. Have you looked into blood-brain barrier permeability in the mouse model with infection? Is it just CL2 driving infiltration, or also loss of blood-brain barrier integrity allowing monocyte entry?
Right. Okay, wonderful question. I think I'll make some notes so I remember all parts of it. So it's not just CCL2 that helps to bring these cells into the brain. So we're trying to parse out what elements of cell recruitment the chemokine is directing the most. So if we block the adhesion molecule interactions, so if we block the interactions with VLA4, so if we block VLA4 or LFA1, we can limit monocyte entry as well. So the adhesion to the endothelium is also important. And then we're not sure if the IL-33 signal is affecting astrocyte end feet, but there's definitely a loss in sort of the continuity of coverage of the neurovascular unit by astrocyte end feet. We don't fully understand that process. We get really, as you may have noticed, the astrocytes are so activated in this system. The blood-brain barrier is not completely wide open, but we can get a lot of large molecules into the brain with inflammation. And so anyway, it's a good question, but just having the blood-brain barrier be open isn't enough to cause these cells to infiltrate. There's still a selection that seems to be dependent on chemokine and adhesion that's also required.
Our next question, did you see immune responses during infection in astrocyte leading to pro-apoptotic change? And do the infected astrocytes still communicate with neuronal cells? And any specific marker in that case?
Okay, so that's sort of coming back to the same question of trying to understand why these cells are dying, right? So one of the interesting things about IL-33 as a molecule is that it does have caspase cleavage sites. So the idea is that a normal apoptotic cell death would end up cleaving the IL-33, so that would be inactive. So this is one of the reasons why we think it might not be an apoptotic release. And so, we don't know what the cell death is, but it has to be a way that gets functional IL-33 out into the cytosol. And whether or not these astrocytes are still supportive of neurons, that's a really complex topic, and a lot of work by Emma Wilson's lab from UC Riverside has tried to address this. The astrocytes become they're not functioning as well as they used to. So they don't tend to mop up as much glutamate anymore, which is a big role that they do to help maintain neuronal function. It gets more complicated than that, but essentially astrocytes are somewhat dysfunctional, and the idea is that leads to increased seizure in mice that are quite sick. So this could be what causes seizure in humans once they present with toxoencephalitis, which is like the really severe form of disease. And so so there's some indication that these astrocytes are dysfunctional, but it's whether or not that's being driven by IL-33 or just the global inflammation, we really don't know yet.
Okay. Next question. Is IL-33 receptor expression on astrocytes required for proliferation of T cells and production of interferon gamma? Are the effects of astrocytes in supporting T cells direct or indirect?
Oh, those are great questions. I hope you've read the paper because that's in there. So this paper was published in PLOS Pathogens at the end of October, which has more details. So yes, the T cell compartment is affected. So there's decreased number of T cells. They make a little less gamma. They don't proliferate as much. Then trying to understand how that's all happening, because it still happens when the astrocytes are ST2 deficient. So this is a bit of a question that we don't really know what's going on. But so when we're thinking about astrocytes that aren't able to respond to IL-33, we think that's a very brain-centric, brain relevant site where IL-33 would be acting, and we still have reductions in T cell responses. So in some ways, this suggests that astrocytes are involved in some way in maintaining the T cell response, but we don't know if that's direct or indirect. We don't have really much evidence at all that the T cells are able to respond to the IL-33 directly either. So IL-33 seems to be important for maintaining the T cell response, but exactly the mechanisms, I think that's a whole new area of research that's going to come out of this.
Next question. Even as meningeal resident immune cells are far away from deep tissue, could there be a role for cytokine production by meningeal resident immune cells that will reach the sites of parasite replication via the CSF?
Yeah, that's a great question. Yeah. Because we're sort of hypothesizing that it's local release of the IL-33 that matters, because it seems IL-33 seems to be regulating the size of inflammation that surrounds where parasites have reactivated. But we don't really have any evidence to exclude the possibility that some of these ST2 expressing cell types within the meninges could be reacting. One of the things that we don't really know is the cell types that express ST2 often make sort of type 2 immune effectors. And so they really wouldn't be very potent in helping to eliminate toxo. So when really thinking about what type of inflammatory mediators and cells that need to reach the deep brain to control toxo, we really think about interferon gamma and things like TNF. And so whether or not we could initiate sort of an ILC response, or if we're affecting Tregs or mast cells, I don't know how that would influence the parasite. And parasite growth. But that is something we're kind of interested in. One odd thing is that in our chronically infected mice, we can't find ILC2s anymore. We find them in the naive animals, but they're gone and once we have infection. And so that has been reported in high interferon gamma environments, but so we're not sure what's going on there, but that's still a great question as to what's going on in the meninges.
All right, and our last question. Considering that interferon gamma is important for toxogondii control, do you know whether the infiltrating macrophages or the microglia cells are producing type 1 or type 2 interferons?
Yeah, so as far as the entire toxo field hasn't focused on type 1 interferons much at all. Early studies suggested that there wasn't much of a role for type 1 interferon, at least during the first 2 weeks or so of infection, but it could be revisited whether or not we have some role for type 1 interferon in the brain. A lot of the type 2 interferon, so that's going to be the interferon gamma. We only have evidence that that's coming from the T cells after an exhaustive look. There have been some reports that the microglia are able to make interferon gamma, but that's only in SCID mice during a really short window of time. So we're not sure how relevant that is to mice with an intact adaptive immune system. So yeah, we think that a lot of the interferon, or all of it's really coming from the T cells.
All right, well, thank you, Dr. Harris. That was a great talk. I'm going to transition over now to my co-president, Natalia Benavides, to introduce the next speaker.
Thanks so much.
All right. Hello, everyone. As Lauren mentioned, I'm Natalia Benavides. I'm also the co-president of EPA ASM student chapter. I'm a third-year PhD student at Thomas Jefferson University, and I'm going to introduce our next speaker. So our next speaker is Ron Lucarelli. He's a PhD candidate from Temple University. He's working under Dr. Vincent Tam. And Ron today will be presenting his exciting talk titled sorry, titled Eicosanoid activated PPAR alpha inhibits NF-kappa B dependent bacterial clearance during post-influenza superinfection. All right, Ron, take it away.
Good afternoon, everybody. My name is Ron Lucarelli, and from the Department of Microbiology and Immunology in the lab of Dr. Vincent Tam. Thank you for the opportunity and the honor to speak to you today about PPAR alpha inhibiting NF-kappa B dependent bacterial clearance and superinfections. Let us begin. Sorry, I'm just going to move this real quick. Secondary bacterial infections or superinfections are post-influenza infections that are associated with increased instances of pneumonia or death. Their historical significance even dates back to the 1918 influenza pandemic, pictured here, Camp Funston, military base in Kansas. And there was an instance of, from historical narratives looking at military bases across the country during the height of the pandemic, where soldiers were getting the influenza virus, as shown here with the blue peak, and seeming to recover. And the following week, shown here in gold, we were seeing they were taking a turn for the worse and having high morbidity or even high mortality outcomes. And what we know from that now is that a secondary pneumonia infection has potentiated and is a contributor to that morbidity and mortality. So it's been suggested around 7 to 10 days post-infection, the resolution of the viral infection helps potentiate the secondary bacterial infection. So when we consider inflammation and superinfections, we have to consider the role of bioactive lipids. So looking at microbial insults or injuries, when they begin, arachidonic acids are metabolized via the cyclooxygenase and lipoxygenase pathway via prostaglandins and leukotrienes, responsible for vasodilation, extravasation, and recruitment of immune cells. This pathway even helps convert metabolites such as DHA to EPA, bringing rise to lipoxins, resolvins, protectins, and myrisins to help return the inflammation back to homeostasis. These can be targeted. Some examples just from everyday life, NSAID relievers like Advil target cyclooxygenase pathway, while asthma treatments can target the lipoxygenase pathway. The third family of metabolites that we're interested in is the cytochrome P450 metabolites, shown here in blue. And we're very interested in them in the context of superinfection. So up here showing when we started doing experimental infection studies on mice, Black 6 wild type mice, with singular infections for staph, influenza, as well as the influenza and Staph aureus superinfection, all the mice will succumb to the superinfection within 13 days from the onset. And when we collected the bronchoalveolar lavage from these mice and looked at lipidomic studies of the different metabolic families, we seen exclusively just about the cytochrome P450 family present during superinfection. Not only that, it's abundantly higher than what we see in either of the singular staph or influenza infections. These metabolites interact with the immune system through nuclear receptors and transcription factors. A major component of that is the peroxisome proliferator activated receptor, or PPAR, a tri-subunit receptor with an alpha, beta, delta, and gamma subunit. What happens is these metabolites serve as a ligand and bind to PPAR alpha or one of the other subunits and serve as the ligand to activate and affect lipid metabolism and immune response as a transcription factor. So initially we wanted to understand, how does PPAR play a role in the outcomes of superinfection? And we set up an infection study with mice with Black 6 wild type mice and PPAR alpha knockout mice. And what we found from that is that knocking out PPAR alpha is actually protective during superinfection. 60% of the mice do survive the infection compared to none of the wild type. And PPAR is highly expressed from an immune context in macrophages specifically and has been implicated in polarization. So we also examined that. And specifically in superinfection, we noticed something very interesting. There's a significantly higher population of M2 macrophages present during superinfection in the wild type mice that is not present in the PPAR alpha knockouts. And M2 macrophages, of course, would be interesting given they're associated with being more anti-inflammatory. Not only that, Using quantitative PCR, we notice there's a dampened inflammatory response in a multitude of genes, including IL-6, IL-1 beta, CXCL5, MMP9, and MARCO in the wild-type superinfected mice compared to PPAR alpha. When we started to look at the lipidomic response with the help of Dr. Joris Beld at Drexel's University College of Medicine, we also noticed some differences in the lipidomic responses. Precursor metabolites such as arachidonic acid and DHA were both significantly dampened in the wild-type superinfected mice, as well as for pro-inflammatory associated metabolites 9-HODE and 15D delta PGJ2. So we decided to return back and look at the polarization question a little bit closer. And we used bone marrow-derived macrophages in vitro experiments where we stimulated with the following conditions to create the polarizations. And then using a chemical agonist known as WY14643, we activated PPAR alpha activity with that chemical agonist. So looking here at the first bar graph that I have, with interferon gamma and LPS, we've stimulated M1 macrophages using interferon gamma and LPS. What's interesting though is when PPAR alpha was activated concurrently with it, there is a significant decrease of that M1 population. It doesn't seem to be exclusive of being an M1 to M2 switch though. With M2A stimulated macrophages using IL-4 and IL-13, we also see the same concurrent reduction of that population when PPAR alpha is activated. It was actually conversely different with the M2B population where we used immune complex and LPS. When PPAR alpha is induced, it actually astoundingly increases the population, nearly triple, a significant change, that there's more M2B macrophages when PPAR alpha is activated. We found this to be quite interesting given the presence of immune complex due to when singular infected mice with influenza about 7 days later in the presence of immune complex have antibodies to influenza. So the M2B macrophages producing IL-10, it would seem that that would be the anti-inflammatory environment that might be driving resolution phase. And concurrently during superinfection, the secondary bacterial infection seems to hijack that and use that to its advantage to establish itself and invade. We further validated this using quantitative PCR, in which we used IL-12 beta transcriptional response for M1 macrophages, as you could see in the black versus red. So this is just black 6 macrophages versus the activated with PPAR alpha induction as well. That population, the transcriptional response is essentially decimated for IL-12 beta to represent M1. Arginase 1 being used to represent M2A, we can also see a significant decrease of that with the IL-4, IL-13 when PPAR alpha is activated. It's only the IL-10 transcriptional response with the immune complex and LPS that when PPAR alpha is induced, that the IL-10 transcriptional response is increased. So based on that, we've noticed that the macrophages are polarizing towards this anti-inflammatory and specifically M2B outcome. We next were interested in how is the immune response being modulated by that. And we did a nanostring encounter inflammation panel in which a variety of immune response genes were fielded with stimulations of a pure cytochrome P450 lipid, 1415 DHET, poly-IC, which is a toll-like receptor stimulant, and then the combination of them both. And on the right, I included some of specific genes to share with you today. IFIT1, IL-12 beta, CCL7, and IL-6. And interestingly, you can see that the activation of PPAR alpha alone with these lipids actually can decrease the transcriptional inflammatory gene response, as seen here with CCL7 from the black to the checkered bars. You could see from black to the gray when the toll-like receptor stimulant is given, there's an increase of all of these as we would expect because they're pro-inflammatory associated genes. But most interestingly and highly significant is the gray to the stripes, which is when there's a toll-like receptor stimulant with the addition of PPAR activation via the lipid. When lipids are given, that activate PPAR alpha, that toll-like receptor stimulation is dampened, and thus it's inhibiting the induction of the inflammatory genes during TLR stimulation. So we next wanted to understand beyond the TLR response is what gene, what is actually being fully repressed during superinfection. And we look to using a motif enrichment analysis in which we use co-regulated genes to see which has the top motif site of repression during superinfection. And what we found from that is that NF-kappa B P65's promoter site is highly repressed during superinfection. So in response to that, we've designed an NF-kappa B reporter assay using, luciferase cell line. I'm sorry, I can't find my pointer. There it is. That we wanted to test and see this concurrently. So unstimulated, there's very little luciferase activity from our cell line for NF-kappa B. And when you give a toll-like receptor stimulant such as LPS, you can see there's a strong luciferase activity. But when PPAR alpha is activated via the direct lipid 1415D hat or the chemical agonist WY, that reporter luciferase activity is greatly and significantly dampened, which further indicate that PPAR alpha induction is antagonistic to NF-kappa B. So in our next portion, we started to look at NF-kappa B translocation more specifically using digital microscopy. And we set this up using an EVOS-2 microscope, and we have bone marrow-derived macrophages that are stemmed with LPS and or the WY to induce PPAR alpha activity. And it's stimulated over 30 to 60 minutes before they're fixed, and then stained with NF-kappa B and PPAR alpha antibodies that we can see using secondary antibodies with Cy5, Alexaflor 488, and DAPI for the nuclear staining. And you get something kind of like this at the bottom. Unstimulated cells are blue with the DAPI. You see a purple from the blue plus the red of the NF-kappa B if it's localized in the nucleus, or you would see a blue-green if PPAR alpha is localized in the nucleus. And we were able to create these bar graphs up above based on our findings of these stimulations. And most interesting to notice when you're looking at NF-kappa B localization, so DMSO being our vehicle control, at the basal level of NF-kappa B nuclear localization, once PPAR alpha is activated within 30 minutes, is dropped by over 50%, a significant change, something we don't see in the PPAR alpha knockouts. You could also see the concurrent increase from the 30-minute point when LPS is given as a toll-like receptor stimulation. And even if you do give LPS to stimulate the toll-like receptor response, to start an immune response, it is severely dampened significantly if PPAR alpha is also activated. And you can see that concurrently with the PPAR alpha nuclear localization here, where you can see there's an increase of localization with just the agonist, but it's still higher than the basal level with even if you give LPS as a toll-like receptor stimulation. So this finally brought us to wonder, how is the macrophage function affected from PPAR alpha induction in superinfections? And we first started off by doing CFU assays with staph infections, where an MOI of 20 was given to the macrophages. And significantly shown in the 30 minutes, we can see that PPAR activated macrophages have a significantly lower bacterial load, shown here in red compared to the black. But we had to refine our experiments further to understand whether the phagocytosis is being dampened or if there's something else at play with the bacterial killing of why there's this difference. So we set up microscopy experiments at an MOI of 20 and an MOI of 50, where we used heat-killed Staph aureus that's fluorescent for M. cherry. And setting this up, what we've noticed is not only the actual count of bacteria per cell here, at both of these MOIs, but also the total area of the macrophage itself that is showing the fluorescence for the M. cherry staph is significantly different and reduced when PPAR alpha is activated, indicating that the phagocytotic functions are actually being hampered by PPAR activation. And as terms for the bacterial killing, we finally set up an experiment with Dr. Tina Butero at Temple University, and she helped us design a confocal experiment to understand what is PPAR alpha and these metabolites doing in regards to the killing functions of macrophages. And so we did that by using fluorescence with cell mask red for the plasma membrane. We have CFP expressing Staph aureus to show the live bacteria, and we added Cytox green to marcate successfully killed bacteria. And what you can see in the first 30 minutes again is that the bacterial killing functions from here, the black to red, are significantly hampered when PPAR alpha is activated in the first 30 minutes. This is a change we don't see within the PPAR alpha groups in the purple and the green. And if you look at the representative images on the left, you can see that in general. There's a mix of blue and green for both the PPAR alpha images, regardless of whether the chemical agonist WI was given. You can see with the wild type, however, the phagocytosis function is still there, but maybe dampened. Also, though, there's not really much bacterial killing. There's not many Cytox positive bacteria present in this image. And this brings us to just an overview of superinfections in general, where we see healthy individuals get influenza. And normally we see a successful resolution and a return back to homeostasis without a problem. But sometimes secondary bacterial infections potentiate, and that becomes a problem because of the abundance of cytochrome P450 metabolites. Those metabolites, we believe through PPAR alpha activation, are inhibiting the classically activated M1 macrophages that would respond to the secondary infection, and instead increasing the M2 macrophage population, specifically M2B, as I pointed out to you. This reduced immune response is seen through decreased phagocytosis and clearance, and the dampened immune response genetically that we are transcriptionally that you've seen as well. And this ultimately causes an immune shock that unfortunately causes high morbidity or even potentially death. And just to acknowledge our lab, Norma Gordachetegui Escalante, our technician, helped a great deal with this work, as well as Jessica Taddeo, our new graduate student. Dr. Nicastro in the Tukel lab and Amanda Miller helped with some of the bacterial work. And of course, Dr. Beld's lab at Drexel and Dr. Butero have been an awesome help with designing some of these experiments as well. And I could take any questions.
Thanks for the great talk, Ron. So we have a question, and it's, for the NF-kappa B translocation data and BMDMs, did you try stimulating with any other TLR agonists, such as TLR3, 7, or even 8?
So we did do TLR3. We used poly-IC as well, and we continued with the LPS, but we have used the poly-IC as well and have had similar results. I don't have them in the slides, but I could follow up with a question in the messages as well if they wanted to discuss that more. I'm like trying to pull up the chat as well so I can see the questions, because it doesn't, I don't think it lets me.
We don't have any other questions. There's still 3 more minutes. If there's any questions, anyone wants to type them in now, or you can also type them in later for Ron separately. There's a question. So
step of your work.
Step for your work. Yeah.
So the next step is essentially, let me see if I have an image that would best like visually go with this. So we are actually in a large part trying to next look at using PPAR alpha antagonists that would essentially get to try in vivo experiments to see if the mice can survive if we are using something to inhibit the PPAR alpha elements of the superinfection. That's where a lot of the work will go, as well as we're going to actually look further into some of the cytochrome P450 metabolites specifically that may be how we could, in terms, block them in certain portions and try to do further studies with the lipidomics. I don't have a visual that would best show that, though. Sorry.
All
Is there any
right.
other so if there's any other questions, you could just message me in the private messenger. Wait,
There's a
did you
question,
investigate
so-
what causes the decrease? And see, I did it. So wait, let me go back to that slide. Decrease of, I think it is the idea that they're being processed by PPAR alpha. They're obviously, we think that they're being utilized in some component that there's such a difference in the amounts present, right? Because if they're precursors, so they may have been metabolized into something else. Perhaps dampening the response, that would be something we would further have to look at. To Lauren's question, that's a very good question. Is there anything else? I think I'm pretty close to my 20-minute point, I think. Am I?
Yeah.
Okay.
All
All right, I will
right,
stop
so
sharing. Thank you very much for your help.
all
And
right,
thanks
so
for
now
listening.
I will thank you, Ron. I'll transition over to Matt, who will introduce our next speaker.
Thanks, Natalia. So please join me in welcoming our next speaker, Hagar Mohammed. She's a PhD candidate in the lab of Dr. Fred Krebs at Drexel University College of Medicine, and she will be presenting her talk, Exposure to Non-Thermal Plasma Increases Markers of Immunogenicity in a Model of Latent HIV-1 Infection.
Thank you, Matt. Okay, can everybody see my screen all right?
Yep, you're good.
Okay, good afternoon, everybody. Today, I want to talk to you a little bit about our work towards the development of an immunotherapy for HIV-1 infection using a novel technology called non-thermal plasma. First, I would like to provide an overview of the immune response to viral infections as a refresher. So when a cell becomes infected, it's able to emit various damage-associated molecular patterns or DAMPs that serve to recruit antigen-presenting cells and promote their function in various ways, as through maturation and phagocytosis. And following this, antigen-presenting cells can then stimulate the adaptive immune response, and in particular, CD8 T cells are really important for viral infections. And these cells can then go on to target and eliminate the viral infected cells and clear infection. So this overall process is usually very successful for a variety of viruses and viral infections, but for some, there can be dysfunctions at any of these stages, which overall cause the individual to have a huge problem with controlling infection. And this is particularly the case for HIV-1 or human immunodeficiency virus type 1. Even today, HIV-1 is a global health burden and still causes lifelong infection for at least 38 million people worldwide. Now, the majority of these people are on antiretroviral therapy, which is a combination of drugs that functions to inhibit viral replication in these infected individuals. And thus far, it's been great at preventing loss of CD4 T cells, and it's prevented progression to acquired immunodeficiency syndrome, or AIDS. So when patients get on ART, let me switch over to the laser pointer here. When patients begin ART, they're able to decrease their circulating virus or viremia to undetectable levels. So the infection never quite gets completely eliminated, and this is because HIV-1 is able to establish latent infection. So if a patient stops taking ART, there can be HIV-1 viral rebound, which means that there will be uncontrollable replication once again. And this, as I mentioned before, is due to weak immune responses that are mounted against HIV-1 that cause patients to fail at controlling infection if they are not on antiretroviral therapy. So There are many reasons for this, and ultimately there are different dysfunctions that HIV-1 patients present with in both their innate and their adaptive immune system. On the innate side, there have been many reports about different ways that antigen-presenting cell or APC function has been impaired. And this is partly because infected cells can sometimes not efficiently produce type 1 interferons or other pro-inflammatory cytokines that are necessary in promoting recruitment and other functions of the APCs. So incomplete maturation as well as impaired phagocytosis has also been reported. On the adaptive immune system side, CD8 T cell effector function has also been heavily impaired in HIV-1 infected individuals that can control viral replication. And this is partly because for latently infected cells, they're not producing viral antigen to be recognized as infected cells and eliminated. And even if they're producing viral antigen, sometimes there's the issue of decreased MHC1 display, which also limits recognition of these infected cells. And of course, epitope escape mutations also greatly contribute to unstable binding to infected cells and subsequent effector function of CD8 T cells. And of course, any dysfunctions that are associated with the innate immune response are going to affect the adaptive immune response because it's necessary for initiating the adaptive immune response. So given that there are so many different types of dysfunctions in HIV-1 infection, we wanted to know if there can be a novel immunotherapy that can resolve these different issues simultaneously and maybe avoid the need for lifelong ART. So for this approach, what we're proposing to do is to utilize non-thermal plasma technology. And we're interested in non-thermal plasma because it has been applied to other disease models, particularly cancers. And it's been shown to stimulate this very, very similar immune response through a variety of mechanisms, through promotion of antigen-presenting cell function by causing release or display of various damage-associated molecular patterns in cells when they didn't do so otherwise. And ultimately, it has resulted in a strong CD8 T cell immune response in vivo in some cases as well. So our hypothesis is that application of non-thermal plasma to cells in HIV-1 infection might do something similar in that it will enhance or modulate the immune response at multiple steps here to overcome some of the challenges that that HIV-1 individuals are presented with. So what exactly is non-thermal plasma? I'm sure a lot of people have no idea what I'm talking about at this point. Well, I'm actually talking about the fourth state of matter. So it is an ionized gas and it's composed of a whole bunch of things, as you can see here. But the main species that are associated with its biological effects are the reactive oxygen and nitrogen species. I'll be referring to these as RONS. And when these species are delivered to cells during application, they induce oxidative stress. This oxidative stress is what is associated with the immunomodulatory potential of non-thermal plasma that's been observed in studies both in vivo approaches and ex vivo approaches in therapeutic models. So in simple terms, non-thermal plasma functions as a method of delivering a controllable amount of RONS to cells to mediate these effects. And we would like to then apply this to HIV-1 infection. So for our model, we use the KinPen device to deliver plasma to cells. And as you can see here, this is the usual setup for how cells become exposed. This was gathered by two of our co-op students in the lab. And during exposure, again, the cells in culture are receiving or being exposed to a variety of reactive oxygen nitrogen species that are going to then cause oxidative stress. So the cells that we used for our studies thus far are JERCAT cells and JLOT cells. JERCAT cells are CD4 T cells and JLOT cells are JERCAT cells with an HIV-1 genome. And for our purposes, they model latent HIV-1 infection. And when they're stimulated, when viral gene expression is stimulated in these cells, they're able to produce GFP as a readout. And here's just a construct of the genome that's present in these cells. And we initially did cytotoxicity studies to see if there's a difference in how these JLOT cells and JERCAT cells are responding to non-thermal plasma. And even at a high, which is what we consider 15 seconds of exposure here, there doesn't seem to be that much of a difference between the response between JLOT cells and JERCAT cells. So we went on to assess how these cells are responding with respect to the damage-associated molecular patterns that they could be emitting in response to NTP. So first, we looked at some pro-inflammatory cytokines that are key to promoting APC function. And we noticed after exposure to non-thermal plasma overall that pro-inflammatory cytokines are released, in particular for JLOT cells, more so than for JERCAT cells, which was a bit surprising. And in particular, IL-1 beta, TNF alpha, and CCL2 and interferon gamma were more increased in the JLOT cells. So this suggested to us that there might be an influence or a contribution from viral gene expression in these cells, which was not expected, but we decided to test for this anyway to see if non-thermal plasma was affecting latency. And we were surprised to actually see that there was an increase in the GFP positive cells, again, corresponding to cells in which viral gene expression was stimulated in the JLOT cells. And there was also an increase in the overall GFP intensity in these cells as well. So surprisingly, non-thermal plasma in some way stimulated viral gene expression in the JLOT cells. So they no longer behaved as quote unquote latent. This has been astonishing thus far, and we went on to investigate other markers of immunogenicity that are associated with promotion of antigen presenting function and an immunogenic phenotype. So what we looked at next were the display of markers that are known to promote phagocytosis by antigen presenting cells. So these markers are calreticulin or CRT and the heat shock proteins HSP70 and HSP90. So we assessed display of these markers on the JLOT cells following non-thermal plasma exposure and saw that there was a great increase in the population that was positive for these cells for all three markers. We also noticed that in particular, the population or cells that were displaying these prophagocytic markers were the ones that in which there was also GFP expression, meaning that these were the ones in which viral gene expression was stimulated, as also can be seen here. It was a pleasant surprise to see that this also corresponded with enhanced or efficient phagocytosis by THP1 macrophages. And this also occurred in a dose-dependent manner. So majority of macrophages in co-culture with JLOT cells were able to phagocytose these cells. So thus far, we've looked at this area of the antiviral immune response and how this could potentially be affected by non-thermal plasma for HIV-1 infection. Another consideration we had is whether non-thermal plasma can modulate surface markers to also affect direct CD8 T cell targeting on HIV-1 infected cells. So we wanted to assess this further as well. So to do so, we particularly looked at MHC1 expression, and we saw that the JLOT cells, in the JLOT cells, there was an increase in the MHC1 expression following non-thermal plasma exposure, but this did not occur for JERCAT cells. So again, this may be indicative of the influence of viral genes being turned on. And interestingly, we also saw that there was modulation of CD28 and OX40 in these cells, which could present interesting avenues into other ways that antigen presentation can be affected by non-thermal plasma overall. But this observation that MHC1 was slightly increased on JLOT cells in correlation with what we've seen earlier with enhanced or increased viral gene expression made us wonder if there are, if these cells are now displaying novel peptides or more immunogenic peptides because there's more sources for antigen to be displayed now. So this is what we investigated next. And to do so, we performed mass spectrometry. So we isolated MHC1 peptide complexes and analyzed the peptide sequences that were presented. And what we saw was that both JLOT and JERCAT cells were actually displaying unique peptides when they were exposed to non-thermal plasma, but a lot of these were actually just host cell derived peptides. And when we tried to investigate the display of HIV-1 derived peptides, it was very difficult to get a high yield. But we did see that there were 2 novel peptides that were not shared with the untreated control. So overall, this tells us that the MHC peptide pool is altered following non-thermal plasma exposure, but there is some work to do with respect to figuring out what is the optimal time to be checking for the presentation of antigens. So thus far, I hope I have convinced you that non-thermal plasma can stimulate viral gene expression and DAMP emission. So this is for both the pro-inflammatory cytokines as well as the prophagocytic markers. In addition, we've seen phagocytosis of NTP exposed cells, and non-thermal plasma on the innate and on the adaptive immune side with respect to direct targeting by CTLs increases MHC1 and alters the peptide pool. So there is potential for novel peptides and maybe very immunogenic peptides to be presented as well following non-thermal plasma exposure. Now, of course, there's still some work to do with respect to figuring out if non-thermal plasma is a strong influencer of antigen presenting cell recruitment, and this can be better done in vivo to analyze whether APCs can be efficiently recruited and mature in response to NTP exposed cells. And this step is also very critical. So it would be important to also assess in vivo what would these APCs, how well these APCs can cross-present infected cell-derived antigen to CD8 T cells to then drive a protective CD8 T cell response. So what we envision for this application is that HIV-1 positive patients can go through some sort of ex vivo immunotherapy approach in which infected CD4 T cells can be obtained from the patients exposed to non-thermal plasma ex vivo, and when delivered back into the patient, inside the patient, these cells can emit these DAMPs that are very immunogenic and attract APCs and promote other functions of APCs, including phagocytosis. And then antigen can then be cross-presented to CD8 T cells to initiate that protective CTL response. And at the same time, we would hope that there would be stimulation of viral gene expression that would allow direct targeting of these infected cells by CD8 T cells as well. And the overall goal of this approach is to allow for ART-free control of HIV-1 infection. So I would like to thank our collaborators at the Leibniz Institute for Plasma Science and Technology in Greifswald, Germany, for their help and resources with all this work, and everybody in our Department of Microbiology and Immunology for their help as well. And I'll accept questions. Thank you.
Thanks, Hagar. So we have a question from Michelle Kotzer. She says, great talk, Hagar. I'm very interested in hearing more about the MHC class 1 peptide breath post-plasma treatment. What mechanism do you think is playing a role to increase the broader response to HIV-1 peptides, which will result in better T cell responses?
Yes, that is a very interesting question and something that we've been thinking about for a while. So as I was thinking about it, I found a recent article about how some latency reversal agents, or LRAs, and some are obviously more robust than plasma, can actually alter the antigen presentation pathway and generate, more peptides or different types of peptides that can be displayed on the surface. So I think that non-thermal plasma can be behaving in a similar manner that way. With respect to the reason why there was increased MHC1 expression, it could just be that viral gene expression was stimulated and now there's just more antigen to be presented. But yes, we have many speculations for how it's occurring, but we need to figure out, we need to narrow it down a lot. So there could be many mechanisms by which this could be occurring.
And another question from Michelle. It's, if successful, would this approach require a personalized vaccine approach, or could you use irradiated T cells treated with plasma?
If it could, sorry, can you please repeat that? If it could require a personalized vaccine?
Would this approach require a personalized vaccine approach, or could you use irradiated T cells treated with plasma?
Well, we would hope it would be more of a personalized approach because even with, between different patients, the dysfunctions can be different in the immune response. So we think that would be more efficient if it's administered as like the autologous CD4 T cells from the patients can be administered in the form of a personalized vaccine. I think that would be most efficient in addressing the differences in their dysfunctions in the innate and adaptive immune response. But I think it could also be possible the other way around.
Thank you.
Thank you.
So if there's any more questions, feel free to put them in the chat. We have our break coming up.
Great talk, Hagar. Thank you so much. Great
Thank you.
job. All right, it looks like right now it's our time for a short break. I would like to just direct everyone. So we just finished our afternoon session. So we are going to take a quick break and come back at 3:20 p.m. for the late afternoon session, which includes Dr. Melo Silva from Dr. Segal's lab. And then we will finish the day with our PESCA lecturer, Dr. Stanley Plotkin. And then please stick around for our presenter recognition and poster presentation awards. So thank you again for attending this early afternoon session. Let's return at 3:20.
I'm just waiting a few more seconds to make sure everybody's back. Okay. Welcome back, everybody. I have the pleasure of announcing the next speaker today. I am introducing Dr. Carolina Rezende Melo Silva. I apologize if I mispronounced your name, but Dr. Melo Silva is a postdoctoral fellow from Thomas Jefferson University, and they're currently working with Dr. Louis Segal, and they're going to be presenting their talk, Type 1 Interferon Receptor Signaling Coordinates Innate Immune Cells to Control an Acute Viral Disease. Welcome, and I look forward to hearing your talk.
Thank you for the kind introduction. It's a pleasure to have the opportunity to discuss and show part of my postdoctoral studies that involve type 1 interferons. I study not only the receptor, but also the cytokines. And today I'm gonna talk to you a little bit about how type 1 interferon receptor signaling coordinates innate immunity cells to control an acute viral disease. Okay, so here. So I'm going to give you a brief introduction, and the main questions we're going to be discussing today are which host cells must sense type 1 interferons through the type 1 interferon receptor for host protection, and what mechanisms are involved in host resistance to an acute lethal infection mediated by this receptor. So type 1 interferons are intraless genes located in the human chromosome 9 and mouse chromosome 4. They were first described in 1957 as virus interference. So they have a classical antiviral function, but they also have been described to play a role in autoimmunity, chronic immunosuppression, and acute pathology. Multiple viruses encode genes that interfere with this pathway, with the type 1 interferon signaling and response, suggesting that they have all evolved mechanisms to thwart this immune response because it's very important for viral immunology. As I said, mentioned before, type 1 interferons are essential for resistance to viral diseases. And when a cell gets infected, it produces the cytokine, the type 1 interferons, which activates bystander and neighboring cells. They activate the upregulation through the FNR signaling of interferon-stimulated genes, which are defined as a set of genes that are regulated by type 1 interferon sensing. And they have two main functions. One of them is an antiviral effect, which results in directly inhibition of different virus replication steps. And the other one is an activation of innate and adaptive immune responses. So in our lab, our focus is to understand protective mechanisms of type 1 interferons that contribute to resistance to viral disease. And viruses of different families that are important to human and animal health, they penetrate the host through disruptions of epithelial surfaces, and they spread to other organs following a lethal hematogenous route of dissemination. And example of such viruses are smallpox, West Nile virus, dengue virus, rubella, measles. And actually, ectromedia virus, which is our infection model, is the archetype type virus that major virology books use to explain this lymphohematogenous spread of dissemination of virus. And our infection model, ectromedia virus, is a large DNA virus, is a member of the genus orthopox virus. It's a natural pathogen of mice, and the subcutaneous infection experimentally mimic natural infection. And it's an acute lethal model. And C57 Black 6 mice are inherently resistant to this lethal challenge. And several other strains of mice have been identified to be susceptible, like BALB/C, DBA, and others. And also many transgenic mice were identified to be susceptible to this acute lethal challenge. So it's an excellent model for studying viral pathogenesis, basic immunology, and vaccine efficacy. So when the virus breached the skin through microabrasions in the footpad of the mouse, the virus replicates in the skin, and it's drained through the lymph node, and then it reaches target organs like the spleen and the liver through its primary viremia. And then the virus, after replicating and causing necrosis in the target organs, reach again the bloodstream and spreads throughout the skin, forming this pox characteristic rash in humans caused by smallpox, or by smallpox, in mouse caused by mousepox, and in camels caused by camelpox. And susceptible mice usually die from day 7 to day 12 post-infection. But resistant mice, they control the virus, and they do by a series of immune response, sequential immune response that is essential for this control. So in the drained lymph node, it's very important that dendritic cells, inflammatory monocytes, and NK cells accumulate and restrict the virus. It's also very important that NK systemic activation occurs around day 5 and day 6. And also, it has been shown that a cytotoxic T cell response at day 8 is crucial for recovery from this acute lethal challenge. So as you might have imagined, it has been described that if an knockout mice, they are completely susceptible to mousepox. So around day 7 post-infection, they die. And to address those questions mentioned in the beginning of the talk, in the overview, we used mousepox. And I wanted to highlight 3 points for which I think it's relevant for this discussion of the role of the type 1 interferon receptor. First is that extramedia virus can infect a multiple range of cells, hematopoietic and non-hematopoietic, including hepatocytes, fibroblasts, immune cells. And susceptible mice perish from a fulminant hepatitis. So the expression of FNR, which is usually ubiquitous in the whole body, but the specific expression in hepatocytes might play an important role, considering that the mice perish from this fulminant hepatitis. Also, I would like to highlight that at day 2 post-infection, the inflammatory monocytes and the dendritic cells that are accumulated in the drained lymph nodes, they are the major producers of type 1 interferon in vivo. So to address the first question, which cells must express FNR for recovery for mousepox, we first used bone marrow chimeras. So we literally radiated either wild type or FNR knockout mice, and we either reconstituted with wild type cells or FNR knockout cells. And we waited 2 months, we infected with Vectormelia. And after challenging these 4 groups of bone marrow chimeras, we observed that all mice that were constituted with FNR deficient bone marrow cells, they succumb to infection very fast. Whereas all mice, or most of the mice that were constituted with wild type cells, they recover, and and the survivors are able to clear the virus. And also we observed that the mice that are deficient of FNR in the hematopoietic compartment, they have increased viral loads in the spleen at day 5 and in the liver in the spleen at day 7 post-infection, suggesting that there is an uncontrolled replication of the virus when the hematopoietic compartment is deficient in FNR. And we further confirmed this information by using a Cree-Lox-P system in which we crossed FNR flux mice with VAV1 Cree-expressing mice. So FNR will be deleted only in the hematopoietic compartment, and these mice are highly susceptible. And we also crossed FNR flux mice with ALB1 Cree-expressing mice which will drive deletion of FNR only in hepatocytes. And most of these mice are resistant, really indicating that even though the replication of the virus in the liver is critical for survival or for susceptibility, FNR expression really is required in hematopoietic compartment and not necessarily in hepatocytes. And FNR expression in hepatocytes might be just secondary. So we further tested within the hematopoietic compartment which cells needed to express FNR. So we know that at day 7, at day 9, at day 8 post-infection, a robust cytotoxic T cell response is important. So we designed a mixed bone marrow chimera system to determine where the intrinsic FNR expression was needed in the adaptive lymphocyte compartment, B and T cells. So we literally irradiated RAG1 mice in which T and B cells would be deficient. They would have no T and B cells in these mice. And we reconstituted these mice, irradiated mice, with 80% RAG1 donors and 20% FNR knockout donors. So in this setting, all the T and B cells would be derived from the FNR knockout donors, and all other cell lines would be derived from the RAG1 donors. So the myeloid and the NK cells, non-adaptive, the innate immune compartment will be wild type, and the adaptive immune compartment will be FNR deficient. And when we challenged these mice, we observed that the survival of these adaptive FNR deficient mice was similar to the control, which was mice that were constituted with RAG1 and wild type cells and would be mostly wild type genotype. And the survivors all were capable of clearing the infection, suggesting that intrinsic FNR expression on a T and B cells is dispensable for efficient adaptive immune response and resistance to mousepox. So our next step was to look into NK cell activation. We also know, and it's well described in the literature, that mousepox recovery is highly dependent on a robust NK cell response. And for that, we crossed FNR flox mice with NKp46 CRE expressing mice. And to our surprise, most of these mice were highly susceptible to ectromelia. And they also had increased viral loads in the liver in the spleen at the 8 post-infection, but not at the 6 post-infection. And NK cells, they are well known to promote that antiviral effect by killing target cells, infected target cells, through granule exocytosis, which results in delivery of perforin and granzyme B and cytotoxic granules into a target cell upon activation of NK cell. So we looked into the expression of perforin and granzyme B in NK cells that are deficient in FNR. And naive NK cells already express some degree of perforin and very low granzyme B, and this is highly upregulated by infection. And so most of the cells are perforin and granzyme B high. And when the NK cells are deficient in FNR, you have a reduced double positive perforin granzyme B, and you have an arrest of a partial population that stays into naive-like levels of perforin and no granzyme B, suggesting that there is an arrest in activation of NK cells when these NK cells are deficient in FNR. And we wondered if this arrest would result in a reduction in vivo killing of infected target cells. And to test that, we infected all these recipient mice, conditioned mice, for 6 days with ectromelia. And at 6 days post-infection, we transferred CFSC label in a 1-to-1 ratio of wild-type cells and TAP1 cells. TAP1 cells, they are MHC low, so they will be preferentially killed by NK cells, even in a naive mouse. And this is further exacerbated by ectromelia activation. So the TAP1 cell population is reduced compared to the wild-type labeled, CFSC labeled donors. And this preferential killing is compromised when the NK cells are FNR deficient, suggesting that FNR signaling on NK cells is essential for efficient in vivo cytotoxicity and target cell killing. Okay, but we didn't fail to notice that NKp46 mice, they are less susceptible than VAV1 mice, FNR deficient mice. So probably there's another cell type that also requires FNR expression. So I also mentioned to you that at day 2-3 post-infection, there's accumulation of dendritic cells and inflammatory monocytes in the draining lymph node. And a previous postdoc in our lab has identified that in the draining lymph node, at that time, post-infection, the inflammatory monocytes not only are recruited, but they're the major producer of type 1 interferons in vivo. And using a GFP expressing extramelia virus, he identified that it's actually the infected inflammatory monocytes that mostly produce type 1 interferons. So we wondered whether the function and this important role of inflammatory monocytes was affected by FNR deletion. So for that, we crossed FNR flox mice with LICE2 expressing CREA mice. And it's well known that LICE2 CREA expression driven LOXP deletion in neutrophils and in monocytes. And surprisingly and very interestingly, we observed that most of these mice that are deficient in FNR only LICE2 expressing cells are highly susceptible to acromegalia. And we looked into the draining lymph node, how the virus replication occurs, and we already found the difference at day 3 post-infection in the LICE2 compared to the controls. But most importantly, at the same time at day 3, the dissemination from the draining lymph node to the spleen has already increased a lot in the LICE2 compared to the FNR flox logs. And also the replication at later points of the virus is also increased in the liver and the spleen at day 5 and day 7 post-infection. But I think the striking reduction of dissemination of this virus by LICE2, by FNR expression, LICE2 expressing cells really shows a picture that the function in the draining lymph node is very important. So to test whether the type 1 interferon production or what will happen with these monocytes when they're FNR deficient, we used a mixed bone marrow chimera approach. So we literally irradiated wild-type mice, and we reconstituted with a 1-to-1 ratio of wild-type cells and FNR deficient cells, and they are all congenitally different, so we can identify two differential CD45 expression, the host and each donor cells. So we infected these mice after 2 months of rest. We got draining lymph nodes at day 3 post-infection. We sorted inflammatory monocytes of each genotype, and we sorted uninfected and infected wild-type and FNR deficient inflammatory monocytes. And by qPCR, we surprisingly noticed that the type 1 interferon production by inflammatory infected monocytes is highly and totally dependent on FNR expression. But I also mentioned to you that the ISG function is also important for an antiviral effect. And we looked also for the virus replication and for the virus replication here and the virus transcription in these infected inflammatory monocytes. And we observed that there is no reduction in the amount of virions being produced, even though the wild-type GFP inflammatory monocytes, they have all the ISG upregulation, which is lacking the FNR deficient, suggesting that in vivo, at least in the inflammatory monocytes, the ISGs mainly are important for type 1 interferon production and other innate or adaptive activation, but not for a direct antiviral effect inside inflammatory monocytes. So just going back to the original questions, and I'm concluding very fast. Now I'm reaching my time. So I would mainly conclude that type 1 interferon sensing to FNR is dispensable or secondary non-epidermolytic cells and in adaptive lymphocytes in vivo. And the type 1 interferon sensing to FNR is essential in innate immune cells, specifically NK and inflammatory monocytes. NK cell cytotoxicity and in vivo killing requires type 1 interferon activation, and that FNR expression on inflammatory monocytes provides an important positive feedback loop for type 1 interferon production. And with that, I'd like to acknowledge all the SIGAL lab and the Department of Microbiology and Immunology and all the resources at SKCC, which gave me all the tools to conduct this work. And thank you for the audience.
Thank you so much. That was a wonderful talk. Does anyone have any questions? Okay, we just got one. And so the question is, were you surprised that IFNAR signaling on T and B cells were dispensable?
Yes, very much. a robust T cell response is essential for extramural recovery. And actually, mice that are deficient in B cells, they die after 2 months of infection because they just can't control without the antibody. So we always have to discuss that point because it's really an important thing. And I know that in vivo, when T cells and B cells, they find type 1 interferons, they would upregulate all kinds of ISGs and they will be activated. However, in vivo, if myeloid cells express FNR, they might produce all the cytokines and all the activation signals that would find a way to activate T and B cells. So they are not exclusively dependent on type 1 interference. I think that's the take-home message. But of course, if you give type 1 interference to T and B cells, they will do something. But I think the take-home message here is that all the cytokines can do the job, not only type 1 interference, but myeloid cells require type 1 interference.
Perfect. I didn't read the second half to that question, but I'll send it to you because we're at time.
Okay.
So thank you so much. I loved your presentation.
Thank you. Thank you for the opportunity.
Hi again, everyone. My name is Marissa Egan, and I'm a third-year graduate student at the University of Pennsylvania. Thank you for sticking around and staying with us for this afternoon session. I am currently the treasurer of the EPA ASM Student Chapter Board, and now I have the honor of introducing our third and final speaker of this symposium. I would like to specifically introduce Dr. Stanley Plotkin. Dr. Plotkin is an emeritus professor at the University of Pennsylvania. Until 1991, he was a professor of pediatrics and microbiology at the University of Pennsylvania, also a professor of virology at the Wistar Institute, and at the same time, he was also the director of infectious diseases and a senior physician at the Children's Hospital of Philadelphia. For 7 years, he was the medical and scientific director of Sanofi Pasteur, based outside Paris. He is now a consultant to vaccine manufacturers and nonprofit research organizations. Dr. Plotkin is also a member of the Institute of Medicine of the National Academy of Sciences and the French Academy of Medicine. His bibliography includes over 800 articles, and he has edited several books, including a textbook on vaccines. He has developed the rubella vaccine, now in standard use throughout the world. He is also a co-developer of the pentavalent rotavirus vaccine and has worked extensively on the development and application of other vaccines, including those against anthrax, oral polio, rabies, varicella, and cytomegalovirus. Today, Dr. Plotkin is also our distinguished recipient of the PESCA Award. Specifically, we would like to thank the PBL Assay Science for generously sponsoring this award. Dr. Sidney Pesca was the founder and chief scientific officer of PBL Assay Science. He was also a biochemist and a geneticist as an emeritus professor of the Department of Biochemistry and Molecular Biology at the Robert Wood Johnson Medical School of Rutgers University. He passed away, sadly, in 2016, and PBL Assay Science sponsors the Sidney Pesca Lecture every year at PIF. So today, we're pleased to honor you, Dr. Plotkin, with this award. A plaque from PBL Assay Science has been sent to your house representing this award, Dr. Plotkin. We hope that it's arrived in time. Dr. Pesca's findings in the field of interferon research and in other scientific disciplines have had a profound impact on all aspects of science, from biochemistry to protein purification and analysis, immunology, virology, cytokine biology, and human health and disease, including the use of interferon as antiviral and anti-cancer therapeutic agents. So without further ado, now we have the pleasure of listening to Dr. Plotkin's talk titled The Past, Present, and Future of Vaccines. Please welcome Dr. Stanley Plotkin. And I will...
Go ahead. So Marisa, thank you very much for the introduction. And I appreciate the PESCA Award. And I will now give a summary, so to speak, of roughly 220 years of vaccinology, which of course means that I will leave out a lot. But let me emphasize what I think are important points. So let's go back in time. Next slide. We are all, of course, very concerned with COVID-19, or the epidemic caused by the SARS-2 coronavirus. But you know, that is not the first great pandemic. If you look at this list, you can see that even going back in time to the plague of Athens in 430 BC, that, and that's probably not the first one, there has been a number there of epidemics, bacterial and viral, that have affected humanity. And I would like to point out the very last line of this slide, the 21st century, and to emphasize that things are getting worse. If you think of the fact that Ebola, HIV, swine influenza, chikungunya, Zika, and SARS-2 virus have all caused epidemics, one realizes that because there are so many of us in the world today, and because of travel, that epidemics are bound to occur in the future. So vaccinology has an important future as well. Next slide. So going back to smallpox, you see this terrible picture of a child affected by smallpox. Hundreds of thousands of people each year would die of smallpox before the vaccine. Next. And as you all know, Edward Jenner at the end of the 18th century observed that milkmaids were resistant to smallpox. He was actually not the only one who made that observation, but he thought about using cowpox to immunize people against smallpox. Although actually we think today that it was a strain of horsepox based on sequencing of the organism. But at any rate, that was a tremendous discovery and controlled smallpox. And it was even important in the American Revolution because George Washington used it to protect his troops. Next. There was a gap until towards the end of the 19th century when Louis Pasteur began to work with viruses and bacteria. The key observation that he made was based on a culture of bacteria that he had left behind during a summer vacation. And when he came back and tried to kill animals with this culture, they were resistant. And then when he made a fresh culture and challenged them, they were still resistant. And that made him think about what had happened to the bacteria. And he used this, as we all know, for other organisms as well to make vaccines. Next slide. And perhaps the most, well, not the most famous, because rabies was the most famous vaccine, but the first controlled experiment was done with his anthrax vaccine, which was simply anthrax organisms that had been heated and desiccated. And at a place called Pouilly-le-Fort outside of Paris, he conducted a controlled experiment in which it says anthrax virus, which of course is wrong. The anthrax bacilli attenuated were given to sheep, cows, and goats, and compared to other animals, and both of which were challenged with virulent anthrax, and 100% of the animals to which he had given vaccines survived while all the others died. So that was of course, a key observation. Next. Interestingly, the next major discovery was made here in the US when Daniel Salmon and Theobald Smith killed bacteria and used those killed bacteria to protect animals against challenge. So that was really the beginning of the first classical killed bacterial vaccines. And the discovery was made here in the US. Next. So here is a timeline going through the 18th and 19th centuries. And you can see that there were a few bacterial vaccines, cholera, typhoid, plague, that were made and which worked. They were somewhat crude, but they worked to protect exposed people. Next. Now, moving into the 20th century, so toxoids were developed by a man by the name of Ramon, a Frenchman, who essentially inactivated toxins from diphtheria and tetanus, and was able to protect people against those two diseases. Also, pertussis bacilli were inactivated and used as a vaccine. As many of you will know, Bacille Calmette-Guerin, which was essentially a bovine tuberculosis bacteria that was passaged several hundred times in the laboratory by Calmette and Guerin, became an important vaccine against tuberculosis. And then Max Tyler attenuated the yellow fever virus, which was epidemic at the time in Africa and South America. Also in the 30s, the first inactivated influenza vaccines were made after the isolation of influenza, which was done by Andrews in the United Kingdom. Next slide. So moving along, a key fact, a key discovery was made just after the Second World War, and that was the ability to grow viruses in cell culture. This was done in Boston by Enders and his two students, Fred Robbins and Tom Weller. I actually studied with Robbins myself. So this discovery next allowed for advanced viral vaccines. As we all know, Jonas Salk developed an inactivated cell culture polio vaccine, which was dramatic in preventing polio outbreaks. Next slide. And the oral polio vaccine was first developed by Hilary Kaprowski. I mention that also because Hilary Kaprowski was the head of the Wistar Institute on the campus of the University of Pennsylvania. And I worked with him way back in the 1950s and 60s. So we can take some credit at Philadelphia for OPV. But next slide. Of course, the licensed vaccine was developed by Albert Sabin, who was better at weakening the polio virus. Next. So without going into details, this gives you the impression of what vaccines were developed towards the end of the 20th century, based primarily on cell culture, but also for bacteria, simply the isolation and attenuation or an activation of bacterial capsular polysaccharides allowed for many important vaccines that are routinely given to children today, and which prevent an enormous amount of bacterial meningitis and other infections. When I was an intern we saw a lot of this, but fortunately, it is gone. Next. So what about genetic engineering? Well, the story goes that Herbert Boyer, and the next slide, Stanley Cohen, were together at a meeting on molecular biology, and they went to lunch at a delicatessen. And in the delicatessen, they had the idea, the mutual idea, about using genetic engineering to produce things using molecular biology. And this, of course, has been extremely important for vaccine development, including, of course, next slide, the hepatitis B vaccine, which was the first recombinant vaccine. But other vaccines have been developed using molecular biology. And as we come to the end of the 20th century, you can see that many bacterial and viral vaccines were developed using cell culture and genetic engineering and protein chemistry to conjugate capsular polysaccharides. And the next slide shows that since 2000, we continue to develop new vaccines for a variety of diseases. I'll just point out one, and that is the zoster subunit vaccine, because that's a vaccine that depends purely on a single glycoprotein, and it's highly effective. So we are using purified entities, purified proteins to produce vaccine. The same thing is true for meningococcal group B vaccine. Next slide. So, as you know, vaccines have been tremendously successful in preventing disease. And if you will pardon me for choosing one of my own, this was the effect of the pentavalent rotavirus vaccine in the US. It was introduced in 2006. The bumps on the bottom indicate confirmed rotavirus infection, and you can see how the disease has practically disappeared from the US and other countries. So that gives you a sort of a broad background as to how vaccinology has been conducted in the past. But now let's talk about the future. Next. So what strategies are being used for vaccine discovery? And of course, I will come back to this in talking about COVID-19. But we still use attenuation to develop organisms that can be used as vaccines. Temperature sensitivity has certainly been used a lot to isolate organisms that are less virulent. An interesting approach, which is being used a lot now, is codon de-optimization. In other words, you take the genetic code for an organism, and as you know, the code can vary for different amino acids. And if you change the coding for particular amino acids, you actually can attenuate the organism. There are other methods like microRNA insertion that are also being used. The next slide. On the inactivated side, of course, one can crudely use formalin to inactivate an organism. But now we are using DNA technology, DNA plasmids, coding for important antigens of both viruses and bacteria to make vaccines. And as anybody reading newspapers knows, we're using messenger RNA and also self-amplifying RNA, which is a different way of presenting the RNA. And they are obviously revolutionizing certain aspects of vaccinology. So-called reverse vaccinology is simply using the genetic code and taking it apart and testing different parts of the code to see what they code for, and then finding the antigens that are coded for that are protective. So it's a way of analyzing bacteria in particular. Looking at what, in particular, bacteria transcribe is useful, and structural biology has really taken off. I think I will mention this in relation to respiratory syncytial virus, but there are other places where structural biology has become important. And new adjuvants are now extremely important in vaccines. Next slide. So let me talk about a few particulars, starting with influenza, because as we all know, influenza is from year to year epidemic, and we use vaccines that, if we're honest, are really not that great. They're moderately effective, but certainly far from 90 or 100%. Next slide. Well, actually, the first part of this slide repeats what I just said, that we have both live and inactivated vaccines, but the inactivated vaccines are less effective in children and the elderly, and the live vaccines are totally ineffective in the elderly. And both types are less effective because the circulating virus mutates and changes from year to year, and vaccine efficacy is only moderate. Next slide. Right, well, so how can we improve the efficacy of influenza vaccines? Well, a number of ways are possible. One thing that's been done already is to use a second lineage of type B. In other words, instead of trivalent vaccines, AA and B, we now have quadrivalent vaccines, AA, B, and B. Increasing the hemagglutinin dose from 15 micrograms is actually very successful in improving efficacy. And we are now using adjuvants. MF59 is oil and water, but ASO1 and flagellin are TLR agonists. So these are definitely improving the response. Neuraminidase is the protein that permits influenza virus to exit the cell. And it's never been used really as part of the vaccine. So we should start to add neuraminidase. And there are other strategies like prime boost, but I want to concentrate in the next slide on conserved epitopes. So here is a cartoon of the influenza hemagglutinin. The red on top is the head, and the blue is the stalk or the stem. And the head, as you probably know, mutates a lot from year to year. So although we do use it to vaccinate, it is not the ideal vaccine. Fortunately, the stem or stalk, there are only 2 varieties in all of the different hemagglutinin types. And so the obvious thing is, well, let's try to use the stalk antigens as well as the head antigens. Next slide. So a lot of attempts are being made to do that. And here are 2 approaches. The one at the bottom is sort of the obvious one. You take off the head and just use the stem. But unfortunately, that doesn't seem to work too well. What does seem to work better is illustrated on the top. and that is to use two different hemagglutinins with the same stalk. And what happens is that the person responds somewhat to the first hemagglutinin, which could be a hemagglutinin of no interest to humans. It can be of something that infects animals, and develops some response to the stalk. But when you come back with a second virus, because the individual is sensitized to the stalk antigens, there's a better response to the stalk after the second injection. So we hope that this will allow us eventually to use stalk antigens. Next slide. Now, I mentioned pertussis. Some of you will know that the old vaccine invented early in the 20th century was simply inactivated bacteria. And that worked, but it was quite reactogenic. So because of complaints, scientists went back to the laboratory and tried to develop vaccines that lacked some of the substances in bacteria in Bordetella pertussis that causes reactions. And so they came up with 1 to 5 antigens, purified antigens, that could be used to immunize against pertussis. Pertussis toxin, fimbrial hemagglutinins, pertactin, and so these vaccines were made and substituted for the whole cell. And lo and behold, that reduced reactions and everybody was happy. But, next slide. Well, pertussis is a serious disease in newborns. It still occurs in children and adults, but it may kill newborns. But what happened was that replacing the whole cell pertussis vaccine by acellular vaccine did eliminate serious reactions. But the disease became resurgent in many countries because immunity wanes after the acellular vaccine. And that was not realized when they first did the original tests. Next. An illustration of this is a follow-up study done in Wisconsin. And you can see from 75% efficacy in the first year, it faded to 12% after 3 or 4 years. So obviously, something has to be done. Next slide. And there are a number of improvements that are taking place and should alleviate this problem. One point is that some of the antigens being used in the acellular vaccines are actually old, that is, from bacteria that were circulating 20, 30 years ago. So the obvious thing is to use antigens from circulating organisms today. But beyond that, adding stronger adjuvants to stimulate not only antibodies, but T cells, Th1 and Th17 T cells, and T follicular helper cells to get better antibody responses. Another important change, instead of using pertussis toxin that's been detoxified with formalin, to use genetically inactivated pertussis toxin, because the formalin actually kills 2 important epitopes on the pertussis toxin, which are responsible for a lot of efficacy. So that's another important step. And then there's the possibility of adding other virulence factors, purified virulence factors from pertussis, Bordetella pertussis. Lastly, and one that is very active at the moment, is to use a live attenuated Bordetella pertussis to boost immunity after initial sensitization with the killed antigen, and that could induce mucosal responses, which would be protective. Next slide. So now let me talk about one of my favorite viruses, cytomegalovirus, and to illustrate a strategy that looks very, very interesting. Well, first of all, the problem with cytomegalovirus is that when it infects pregnant women, if they're seronegative, and sometimes even if they're seropositive, the virus can go to the fetus, and the fetus, once infected, can be born with chorioretinitis, hearing defects, microcephaly, all kinds of bad things. So obviously we need a vaccine. But the problem is that this is not measles or polio. That is to say that those diseases are viremic diseases, where the organism passes free of cells in the bloodstream to organs which it damages. So if you put antibodies into the bloodstream, you prevent that. Very simple in principle. But cytomegalovirus is not like that, because it gets rapidly into cells in the nasopharynx where it infects. And from there, the cells can travel to other organisms. And of course, at that stage, antibody is not going to do as much as it would for viremic organisms. So that's a key issue. Next slide. So what is being done to develop a vaccine? Well, this cartoon of the virus illustrates two important things on the surface of the virus in the upper right-hand corner. First, a glycoprotein on the surface called GB, and the second next to it is actually a pentamer with 5 different proteins that are produced by the virus and which come together in a pentamer. The point is that the GB, antibody to the GB, is protective in certainly in preventing acquisition of the virus. And the pentamer seems to be important in preventing the virus from going from a pregnant woman to her fetus. In other words, entering the placenta, replicating in the placenta, and going to the fetus. Next slide. This is just to show this was a experimental vaccine that we developed some years ago. And this is in kidney transplant patients who get cytomegalovirus disease. And if you look at the control population, the central bar, about 35% had severe disease after transplantation due to cytomegalovirus, but the vaccine reduced that to 5%. So it simply showed in principle that you could develop a vaccine against cytomegalovirus. But next slide. But the problem is to get responses to the key antigens which I showed you, and the virus or the vaccine that I just showed you was actually lacking in the pentamer. So the question is how to have a vaccine that has both the GB and the pentamer. Well, the people at Merck had a very good idea. They took 2 proteins of the cytomegalovirus and linked them to a chemical. And what the chemical does, basically, it prevents the code for those 2 proteins to be read. So the virus can't reproduce because it lacks those 2 proteins, the UL51 and the immediate early 1 and 2 proteins, which are shown in the figure. But if you add a chemical called Shield 1 to the culture, the virus that inactivates those two chemicals, and the virus is able to replicate. So the people at Merck can inoculate cell cultures and make these viruses that have these chemicals in them. But then when you inject them into a person who doesn't have Shield 1, of course, then the virus is unable to replicate fully. It replicates in part, and it exposes the human subject to the GB and the pentamer, which are the important immunizing proteins. And this is in clinical trial now, and we hope it will prevent infection and be usable in women or adolescent women and women who plan to have children to protect the child against CMV. Next slide. Now, you know, I hope you know, that there is an excellent vaccine against the human papillomavirus that causes cervical and nasopharyngeal cancers. And I mention this because it's obviously one of the first anti-cancer vaccines, hepatitis B being another one. And a highly effective vaccine, certainly more than 90% effective. Next slide shows those figures. It protects also against genital warts, but against vulval and vaginal cancers, and also against cervical cancers. Okay, well, that's great. But what about women who have already developed cancer? Next slide. And the interesting thing here is that a DNA vaccine is showing efficacy in causing reversion of cervical cancers using the two oncogenes of human papillomavirus, E6 and E7. In the pink, you see the control population of women, and in blue, the vaccinated women. And you can see that there's even clearance of the tumor in most of the vaccinated women. And this, of course, is being developed as a licensed vaccine. Next slide. Respiratory syncytial virus has been a headache. It causes disease in the very young, in infants, and it causes respiratory disease in the elderly, which can be quite serious. Next slide. So as I said, it's the #1 respiratory infection of infants, also important in the elderly, and attempts to develop vaccines against RSV have gone on for many years without a lot of success. Inactivated vaccines actually worsen disease because of immune complex formation, and live attenuated viruses were insufficiently attenuated. Next slide. Then a discovery was made at the NIH in structural biology, which is leading to a lot of interesting work. If you take RSV virus in its, what shall I say, in its native form, it has a fusion antigen on the surface, which is in what's called the pre-fusion state. And in the center of the figure, you see this rounded F protein. And on that F protein, shown on the right, are epitopes that actually cause or induce protective antibodies. But the thing is that what people were doing was putting the virus into cell culture to make fusion antigen. And what was happening was that the pre-fusion was changing to the post-fusion form of the F protein and hiding the important epitopes. Next slide. And well, I won't go into details in this slide, but the antibodies to the prefusion form are protective, and this prefusion F form is being developed to vaccinate both the young and the old against respiratory syncytial. Let's go to the next slide and skip that. And I want to finish by talking about SARS-2, the cause of COVID-19. Next slide. So you probably all know this, but just to make the point that on the surface of the virus, a key protein is the spike protein, usually abbreviated as S. Next slide. This protein, well, I won't go through this, but this protein has important epitopes, including the receptor binding domain that attaches to the angiotensin receptor and is able to get into human cells. Next slide. So how to immunize against this? And the great thing about this, I mean, to make good of a terrible event, is that the vaccinology community has responded by using every known strategy to make vaccines, to make vaccines against SARS-2. Vectors being one of them. MVA is a derivative of vaccinia. Human and chimpanzee adenoviruses are being used as vectors. And even measles virus. Now, you would think that that wouldn't work, but in fact, if you insert proteins into measles virus, you can use attenuated measles virus as a vector. And lastly, vesicular stomatitis virus, which was used for Ebola. Next slide. And the additional vaccine platforms, which you're probably aware of, include simple inactivation, but also changing the genetic code of the virus to make an attenuated SARS-2. I talked about the vectors. Simple protein subunit using the spike protein as the antigen, but also the DNA plasmids and the messenger and self-amplifying RNA. Next slide. Well, I'm not going to go into detail here, but the point is that not only the adenovirus vector is being used as an intramuscular vector for the spike protein, but also as an intranasal vector. And that's interesting because we need a secretory response to eliminate SARS-2 from the nasopharynx. If we can't do that, the virus will continue to spread, even if we're protecting individuals from disease. So moving on, next, I think I'll skip that and go to the next one. And what this emphasizes is the speed with which DNA and mRNA vaccines can be made. This illustrates the DNA story. Using the sequence for Ebola virus, a vaccine was made in 15 months. Then the same outfit made a vaccine against Middle Eastern respiratory syncytial syndrome virus in 9 months, and also against Zika. And then it only took 10 weeks to make a candidate a DNA vaccine against SARS-2. And the same thing, of course, has been true for messenger RNA. And I'll move to the next slide, which simply shows you the responses to Zika DNA, which were excellent, and we can certainly stop the next epidemic of Zika with the vaccines that we will have available. And the next slide talks about both the messenger and the self-amplifying. The messenger has been the easier one to make, and as the diagram shows, you take the messenger RNA for the spike protein, and it is able to enter the cell using a lipid adjuvant, and then is released in the cell and starts to make the protein that you're interested in this case, the spike protein, which can be harvested from the cells and, or rather injected into humans, who will then make the spike protein themselves in their own cells. And the next slide simply shows how good this is in terms of inducing and neutralizing antibodies. This is using the cytomegalovirus proteins as an illustration. At the bottom are the two proteins I told you about, the pentamer and the GB, and you can see the phenomenal antibody responses that are being made to these cytomegalovirus proteins. Let me conclude the next slide, saying there are still unsolved problems in vaccinology, stimulating better immune memory, for example, finding better correlates of protection using systems biology. Next slide. Improving the way we can immunize very young infants and the elderly because of the problems of the changing immune system. The difficulty of using mucosal application of vaccines to induce immune responses, and the importance of more adjuvants. So let me finish with the last slide. Next slide, please. So many of the people who are listening to me, maybe most, are students. And I just wanted to conclude with this. This slide shows a poster from the film made of the novel Arrowsmith by Sinclair Lewis, which is the story of Martin Arrowsmith, who was a young physician who, during the novel, eventually becomes a laboratory scientist. At age 15, I read this book, and it changed my life. Arrowsmith's mentor in the book is Professor Max Gottlieb, who says to Arrowsmith, quote, "To be a scientist is not just a different job, so that one should choose between being an explorer or a bond salesman or a king or a farmer. It is a tangle of very obscure emotions, like mysticism or wanting to write poetry. The scientist is intensely religious. He or she will not accept quarter truths because they are an insult to the faith. So I say to you, search for the whole truth, nothing but the truth, so that it lives after you." Thank you.
Thank you so much, Dr. Plotkin, for that incredible talk and ending on such an incredible note. So thank you for that. I'm just going to stop sharing right now so we can take a look at the chat. Okay, so it looks like there's some questions. So this one is from Dr. Kutzler. She says, Dr. Plotkin, thank you for the wonderful talk today. We really appreciate all of your contributions to the field. I would love to hear your perspective on what you are excited about for the future of vaccine development. What is an example of a current innovation that you think will have a great impact on the future of vaccines?
Well, there are many, some of which I illustrated in relation to COVID-19. Maybe one that I did not sufficiently emphasize is structural biology. Because, you know, when we play with proteins, they change. And what we have not recognized sufficiently is that when they change, their function changes. And so we need to learn a lot more about stabilizing proteins so that they exert a biological effect, which we intend. So I would emphasize that. And of course, you know, I've shown you, again, using SARS-2 as an example, how many different ways there are now to make vaccines, which certainly was not true when I started.
Thank you so much. If anyone else has any other questions, okay, thanks. Lauren has a question. She said, a lot of people outside of science are asking me about future safety concerning mRNA viruses and vaccines. It is possible, is my understanding that mRNA is not super stable and it will either be translated or degraded. How would you answer these people's concerns about mRNA vaccines?
Well, have you got an hour or two? I say that because the subject of vaccine safety is very complicated in a sense. So let me try to summarize very briefly by saying that if we look at the history of vaccines, we have had some negative events like the first rotavirus vaccine caused what's called intussusception. But by and large, the rate of true reactions is very low. So I did a very simple thing recently and actually published it. We have a system in this country of recompense for true vaccine reactions. In other words, if a parent can show that a child has had a significant adverse reaction, they can obtain from the government of the US a monetary settlement, which is considerable. So I ask myself, how many settlements have there been in relation to how many vaccine doses have been given? And it turns out, as a generalization for the vaccines that we're using, it turns out to be about 1 in a million. So that gives you a concrete idea as to what the risks are. Now, the other point that I'll make briefly is that we have a very elaborate safety system in this country for vaccines. We have the VAERS system, which is passive reporting. We have units that do active evaluation of people who think they've had vaccine reactions. We have safety monitoring in places like the Kaiser Permanente system in California, you know, where they have millions of people and they can compare vaccinated people with unvaccinated people. So my answer, my brief answer, is that we have very good systems for evaluating vaccine safety. Now, is it possible, after having given the vaccine, as we're doing now, let's say to 30, 40,000 people, that something turns up in the 100,000th person who gets vaccinated? Yes, but you have to, I say that every child should have a mandatory statistics course because understanding risk is so fundamental. If your risk of getting a disease is 1 in 100, and your risk of a vaccine reaction is 1 in a million, you'd be an idiot not to take the vaccine. But opponents to vaccine like Wakefield, will argue that point. But I've told you what I think.
Thank you for that brilliant answer, Dr. Plotkin. We have another question from Dr. Kutzler. She says, as a vaccinologist, I'm concerned about the vaccine hesitancy rates that are being reported for COVID vaccines. Is there something we can do as a scientific community to help educate, communicate, and build trust of the community?
Well, that's a very important question, and I would be lying if I said that I have a really good answer to that, because, people are not logical. And so all that can be done is to collect the safety data, advertise the safety data. The 3 presidents have agreed to take a COVID vaccine just to show people that they trust the system. And that's about all you can do. People, experience teaches us that most people will accept expert opinion, so to speak. Some people will demand specific answers, and that's fair enough. And then there are people who are irremediable. I mean, they will simply not accept anything you say, and that's nothing we can do about that. In this country, as you know, we have mandatory vaccination for children, and this gets complicated because I have just written a paper about COVID-19 for children. But leaving that aside, you'll never convince all the adults to be vaccinated. It just won't happen. Whether they get sick or not, of course, who knows?
Thank you so much, Dr. Plotkin. Are there any other questions for Dr. Plotkin? Okay. If there are no more questions, I just want to say thank you again, Dr. Plotkin, and congratulations on receiving the PESCA Award. Now I'm going to turn it over to Dr. Kutzler, who will deliver some closing remarks and the presentation awards. Thank you.
Thank you very much.
All right, thank you, Dr. Plotkin, and thank you, Marissa, for moderating such a great session. I'm here to close out a really wonderful 29th Annual Philadelphia Infection and Immunity Forum. I'm just so pleased with how today went, with all of the wonderful presentations from all of our students and postdocs. And I just want to spend a few minutes just with some thank yous and also to deliver our presentation awards today. So first, I'd like to thank our 3 faculty speakers for joining us today: Dr. Taji Harris from the University of Virginia School of Medicine, Dr. Paula Watnick from Harvard Medical School, and again, Dr. Stanley Plotkin from Children's Hospital of Philadelphia. And I'd also like to recognize all of our student and postdoctoral speakers today. What a wonderful job with all of your presentations, such exciting research projects, and answering all the questions that were posed today. It just really made a very rich day across several areas of interest to all of us at Eastern Branch of Pennsylvania and ASM nationally. I know we have a lot of people listening in from across the country. I'd like to recognize Dr. Lubin, Dr. Melo Silva, as well as Rena, Alicia, Ruchika, Ron, and Hager. What a great job that you all did today with your talks. Thank you very much. So now, without further ado, I'd like to recognize all of the faculty that judged posters today during our lunch break. So thank you so much to all of our faculty help for providing scores and really great feedback to all of our presenters. So I've collected all that feedback on our posters, and I'll be providing that to all of our presenters today. And with the scores that came in, I'd like to just congratulate all of our presenters for posters, and I'm going to be recognizing the top top presentations at this time. So I'd like to congratulate the following undergraduate student poster presenters today. First, our first recognized presenter is Emma Keeler from the University of Pennsylvania. Great job. And we also have a second winner shared by co-authors from Rowan University, Cynthia Lacerez and Christina Panko. So Emma's poster was entitled Computational Prospecting of Viral Dark Matter in Search of Adenoviruses. And Cynthia and Christina's poster was titled Inhibition of a Functional Site of Herpes Simplex Virus, Glycoprotein D, through the use of small molecules. So congratulations to all three of you for your presentations today. We also have a medical student presentation award, and it's always wonderful to have our future physician scientists take part in this meeting. And we're recognizing Brandon Rogalski from Drexel University College of Medicine for his presentation, Longitudinal Assessment of Clinical and Immunological Responses to COVID-19 in a Hospitalized Patient Population. So great job, Brandon. We also like to recognize our first and second year graduate student presentation awardees. So these are students who are in the early stages of their graduate careers. And the two students that we're recognizing today, actually, I'm sorry, for this award, we have just one student being recognized, Bhavani Taramangalam from Drexel University College of Medicine, for her poster entitled Adenosine Deaminase 1 Induces DC Maturation and Improved Adaptive Immunity. We also had several MD PhD students, again, physician scientists, future physician scientists at our meeting today. And our awardee for the MD PhD student presentation is Ellen White from the University of Pennsylvania for her presentation entitled Leveraging the Human Skin Commensal Alkylogenes Faecalis to Improve Early Wound Healing. Congratulations, Ellen. We'd also like to recognize our postdoc presentation award, which is going to be given to Arwa Abbas. Dr. Abbas is from the Children's Hospital of Pennsylvania for the presentation entitled Parsing Out Polymicrobial Interactions During Clostridioides difficile Infection. Congratulations, Dr. Abbas. And finally, I'd like to recognize our third plus year graduate student presentation awardees. Our first place prize goes to Beatrice Herman from the University of Pennsylvania for her presentation entitled Defining the Bacterial and Host Factors Responsible for Caspase 111 Independent Cell Death During Infection by Salmonella enteritidis. Our second place poster presentation award goes to Alexander Smith from the University of Pennsylvania for their poster entitled Microbial Cooperation Enhances Clostridioides Difficile Pathogenesis. And our third place poster prize goes to Matt Bell from Drexel University College of Medicine for his presentation entitled Age-Associated Defects in the Adaptive Immune Response to C. diff Infection Impairs the Development of a Protective Immune Response in the Elderly. And we also have two honorable mentions from our third plus year graduate students. The first is Joshua Soto Okana from University of Pennsylvania for his presentation entitled Defining the Impact of Non-Steroidal Anti-Inflammatory Drugs During C. diff Infection. And also to Jennifer Connors from Drexel University College of Medicine for her work entitled The Impact of Aging on Antiviral Pathways and Consequences for Immune Function. So congratulations to all of our poster awardees. And I'm very pleased to announce that our third plus graduate students will be presenting oral presentations at our May monthly meeting. So we look forward to our poster awardees presenting in next May's meeting. And we'll be following up with all of the students to plan that. I'd like to just take a moment again to thank our sponsors from the meeting today. So we're very appreciative of PBL Assay Science for their continued support each year, and in particular for supporting the Sydney PESCA lecture. And also to American Society of Microbiology for their conference grant to help fund this conference again this year. And I'd particularly like to acknowledge the work of our student officers. So it's a very active student branch of the Eastern PA branch of ASM. And our student officers have shown such great leadership this year and their enthusiasm. They are the reason this meeting was so successful. It was our student officers that selected our faculty speakers, invited our faculty speakers, and really put together such a wonderful day of talks across all branches of ASM. So I just want to thank them for all of their work and moderating such great sessions today. So for our co-presidents, Lauren Springer and Natalia Benavides, who's both from Thomas Jefferson, to our co-vice presidents, Matt Bell and Kayla Sakaris from Drexel, our secretary, Ron Laccarelli from Lewis Katz School of Medicine, and our treasurer, Marissa Egan from University of Pennsylvania. You guys did such a great job. So thank you so much for all of your hard work. And I would also like to thank our colleague and webmaster and meeting host, Dr. Vincent Tam from Temple University. He's also one of our faculty members of our executive committee. And I know a lot of our executive committee members are here today. And so I'd also like to recognize our branch and their support of this meeting every year. And I'm just going to end with a plug again for those of you that are not members of the American Society of Microbiology or a member of our branch. It's really easy to become a member. And so if you go to our website, right on the front page, you just have to click become a member. And as Dr. Shefferly said this morning, really becoming a part of this community is there's so many benefits just being part of this community, to network. Also if you're a trainee, a student, just to build your career, and there's just a lot of great science. So I really encourage everyone to become a member of the branch and to stay in touch over the next year. So with that, I'd like to close out the meeting and congratulate everyone on great presentations, and thank you very much for attending today.